Shall we? All right. So good morning, everybody, once again. So we're going to do the final lecture, which is going to basically be a recap lecture of what we've learned. And what we've done is, after this lecture, we're going to be holding a quiz just to kind of, see how well you've understood. It's not meant to be a competition. It's not as if we're going to check your scores and we're not going to do a comparison. It's you versus you. The most important message here is I would like to see if you really had, made some progress in terms of the understanding of the coursework that has been, delivered to you, right? So feel free to, take any notes during this whole recap thing, because most of your quiz is based on what we've learned. There's nothing going to be coming somewhere, under the blue sky kind of questions here. And they're going to be clear multi-choice questions at the end of this particular lecture. Now, this lecture is meant to be a recap lecture because, most of the areas that we've covered as part of the whole GIAN series have been, topics that are completely new, topics that are completely, that are kind of in initial stages. There's a lot of research work going on, and I've been giving you examples from my own research group. So we're going to carry on the same lines, and we're going to look at, again, a recap lecture here, and we're going to go over the whole thing one more time. All right, so welcome to this final lecture, which is on the recap of all the contents that we covered as part of the GIAN series here. So I think one of the things that we learned here is about electrochemical biosensors. We've restricted this whole GIAN lecture series on electrochemical biosensing, right? Because electrochemical biosensors, as we all realized, they're more easy to miniaturize, and then if you can miniaturize things, you can easily implement them in the field settings, right? Rather than having your electrochemical sensors, which are based on huge big machines which either sit in a lab or sit in a big hospital, in a specialized hospital, you need something which can be more point of care, right? Therefore, we spoke about electrochemical biosensors. And as part of this electrochemical biosensors, what we saw here is basically you can have different types of samples where you can use your electrochemical biosensors. You can have cell culture samples, you can have human samples, your blood, urine, and saliva samples, your food samples, or you can have environmental samples. And then you have your biosensor, which is right in the middle of that, which basically would have your transducers, you would have your bioreceptors, and you would have your electrical interfaces. In bioreceptors, you can have different types, which in bioreceptors. In terms of bioreceptors, we could have different types of bioreceptors. We'll cover them a little bit of more detail in the following slides. And then you have your electrical interfaces, which include things like, you've done an experiment yesterday as well. Can anybody tell me what was your transducer yesterday that you used for making your own glucose sensors? Does anybody remember? You had like a flat planar electrode. What was that electrode called? Yes, it was made up of platinum. Well done, good answer. It was platinum. What format was it called? It was in? It was called a PCB electrode, a printed circuit board electrode, right? Something that has been printed there, right? Again, based on printing technologies there. All right, so this is basically some of the things that we've already seen. And then we also saw that we were having an electronic system. You saw there was a potentiostat being used. Was it a single channel or a multi-channel? It was a single channel one, yeah? So you had 4 working electrodes, but you could only measure from 1, right? So it was a single channel electrode. And then you had an electronic system. You were getting your display. You were getting a final value of the current. You were all measuring current over time. What is that technique called? Well done, fantastic, amperometric. I think we're already having the quiz as we progress, and I can already see that you've made a lot of progress here. Well done. So you were basically using amperometric techniques. So you were having a display there, and then in the end you were plotting it and you were getting a straight line. If you got a straight line, well done. That means you got very good sensors. If you didn't get a straight line, then you go back to the drawing board and try again. That's how the whole thing works. So what I mentioned also was at the heart of your biosensor is the bioreceptor. You can have different types of bioreceptors. The main function of your bioreceptor is to provide you selectivity and specificity. I would say specificity is the most important thing. The most important thing with your bioreceptor is to provide you specificity. So you have different types of bioreceptors. You have the enzymatic biosensor, you have the immunosensors, you have the aptar sensors, and you have the molecular imprinted based sensors. And then we saw this particular nice slide on enzymatic biosensors. In the UK, when I teach this on enzymatic biosensors, this is most of the times my first, this is also what I use for questioning students as well. So I would normally say, here's how the glucose biosensor works. You've got the different generations here. You've got the first generation where you're producing hydrogen peroxide. You're using the overpotential to break it down into water, and you release electrons into the external circuit. And then you have the second generation wherein you bring in a a mediator. And then you have your third generation, which is direct electron transfer. You can apply the same thing also to any other oxidase based sensor like lactate oxidase as well. You can have first generation, second generation, and third generation there as well. All right, so that's your enzymatic biosensors. And then the next example we have is that of electrochemical immunosensors. Immunosensors, as the name suggests, immuno, it always relates to antibodies, right? In this particular case, you can see you've got two sets of antibodies. This is more like a ELISA technique that you have here. You have your enzyme linked immunosensing here. You have a primary antibody which has been immobilized, and then you have a secondary antibody which comes from the top. This is more like an enzymatic, or it's more like a sandwich assay that you normally see here. So this is an example of your electrochemical immunosensor here. And then the next example we saw was that of APTA sensors. APTA sensors are basically sensors which have, what do they have as their bioreceptor? Aptamers, right? What are aptamers? Aptamers are basically strands of oligonucleotides. Well done, very well received. So you have your oligonucleotides, and then I mentioned about this very interesting theory wherein thiols can form self-assembled monolayers on gold. All of you are master's students, most of you are master's student or bachelor students or PhD students here. If you know all of this, if you understood all of this, you have actually expanded on your skill sets. It may be a bit of theory here, you've done a bit of practical there, so this is all very nice information that can go set in very nicely into your CV. And if you mention in your CV that you've done courses on wearable technology, you stand much ahead of other people, which allows you to apply for a nice PhD studentship anywhere, either in India or abroad, right? So please, as I said, first of all, I don't have to say anything more. Looking at the response, I don't have to say anything more. You've all received it very well, and you've all, actually focused and done a great job, which is a big satisfaction for me personally. All right. So this was the example of electrochemical APTA sensors here. As you see here, you've got your gold surface, you've got your thiolated aptamers, and these aptamers are self-assembled, and then as soon as you have the binding, you see a change in the electrochemical properties, and that's then measured externally, and that's normally represented here, as you can see here from these electrical impedance measurements that takes place here. Okay, let's have a look at the next one that we have. This one is something that is very, very interesting, and I say that this is the future for tomorrow. I myself am participating in conferences. I'm chairing a conference next week. I'm chairing a session on diagnostics, and there my talk is mainly on molecular imprinted polymers. We are also having this new PhD studentship coming up, so I'm meant to be talking about diagnostics from lab to bedside kind of diagnostics and those kind of things. And there my topic of presentation is molecular imprinted polymers. We've already had, Professor Khalid and myself have had a lot of talk on this particular topic, and this is going to be, I think, the future of tomorrow, right? Because by itself, it gives you a good dynamic range, and you combine this with another receptor, and you can actually increase the dynamic range. You can actually make it more ultra sensitive, right? Because what is it that you need? You need to have sensors or biosensors which are ultra sensitive, which can measure very small quantities of your biomarker. Anybody heard about circulating tumor cells? Yeah, you've heard about circulating tumor cells? Circulating tumor cells, finding circulating tumor cells in the body is like finding a needle in a haystack. That's the kind of challenge there is. And biosensing technologies, mainly molecular imprinted polymers, combined with other receptors, combined with other bioreceptors, seems to be like a technology that might be ready to accept that particular challenge, right? That's why I see a lot of promise on that, and that's why I'll be spending a little bit more time on molecular imprinted polymers to recap on the molecular imprinted polymers. As I mentioned, it's very fondly called as synthetic antibodies or plastic antibodies, and you can tailor the specificity, right? Number one, they're synthetic, they're highly stable. I gave you the example wherein we made devices with molecular imprinted polymers, sent them to Japan by courier. You can imagine they went through all the cold conditions, dry conditions, hot conditions. Japan during summer gets very hot, right? And it went and it survived. It gave us the same results that we were seeing at Liverpool, right? So you need something where you don't require a cold chain. You need something which can survive the extreme conditions. You need something that not just works in University of Liverpool, but also works here in NIT Rourkela, which also works in a lab in Kampala or in Accra or somewhere in, where it's highly humid in Kolkata, right? Or Bombay for that matter, yeah? So you need something that works in all harsh conditions So for that, molecular imprinted polymers seem to be a very suitable candidate, and because you're synthesizing them, you're just putting a drop. Did you realize that you were just adding a drop of the solution yesterday? Did you do your electropolymerization by yourself? How much were you adding? You're adding microliters. I don't think you even went for milliliters. So if I remember very well, a day before, Rishita was preparing solutions which were about 1 milliliter, 2 milliliters. That's it. And weighing in milligrams. So you can imagine, if you can have a drop which can give you a device, you're actually looking at cost-effective solutions here. And you're making them yourselves. It's not as if you're buying them off the shelf, right? And when you buy them off, when you buy things like the enzymes off the shelf and things like that, then you're spending a lot of money. But these ones are much more cheaper. So they're very cost-effective, they're durable, and they have a long shelf life. And then I also showed you how we can make these. I showed you how we have the template molecule in the first step, which is basically your target analyte, right? So you have, let's say for example, p-tau proteins is what you're developing the sensors for your biosensors for. So you start with the p-tau proteins, and then in the next step you bring it in contact with the monomer. For example, it could be phenol red, right? So bring it with the monomer, phenol red, and then you let it interact. There would be interactions at the molecular levels. We also looked into those interactions. Do you remember that? What were those interactions? Anybody? Louder, please. Well done. Hydrogen bonds to stabilize it. What else? Electrostatic interactions. Electrostatic attraction would stabilize it. Electrostatic repulsion would destabilize it, right? So we saw all of those things there as well. And then we also looked at how then you cross-link around it. And in the whole process, when you cross-link around it, what you manage to do is you manage to maintain a particular geometry, a particular configuration, and you not only conserve the geometry, the three-dimensional structure, but you also conserve the charge around it as well. You have those charge centers there, which are ready there to kind of come and bind there. And then in the next step, what you do is you eat away the bioreceptor molecule, or you digest it, or you break it down, and then you end up having these kind of cavities that you see here, where you don't see the green molecule anymore there. All right. And I gave you the example of vancomycin, if I remember rightly, which was the example where we were developing molecular imprinted polymers for vancomycin. All right. And then after that, we also covered a little bit about dual receptors. Sometimes a single receptor does not give you the desired specificity and selectivity. Then in that case, you go and start using dual receptors. And this is another field that I would like to talk a lot about, because I've just attended recently the World Congress on Biosensors in Portugal, right? And one of the big areas which is considered to be the future, right? So this whole talk is not just about, this whole lecture is not just about the recap that we were doing, it's also identifying what is the future and where are the challenges there as well, right? So we'll be trying to do that as well. So if you talk about future, if somebody asks you, if you're sitting in an interview, what is the future of biosensing devices? How do you think ultra-sensitive devices or ultra-sensitive detection can be achieved? You can say, if a single receptor doesn't give you the desired specificity and selectivity and dynamic range, you can combine it. For example, aptamers by themselves probably will not give you the desired dynamic range. And the molecular imprinted polymers by themselves will not give you the desired selectivity, right? So in that case, you combine these two and you get the desired specificity and selectivity. And we've seen examples of that, which I'm going to recap again here. So that's how we covered the whole dual receptors, right? In terms of dual receptors, we have single bioreceptors. Khalid, can we not do it now because my Apple Watch is very sensitive to whatever you're doing there. So we have dual receptors, which can basically, wherein you combine the two recepting systems, and we do this to improve the binding affinity, to reduce cross-reactivity, and most importantly, to enhance signal amplification. And there are several types of dual receptor systems. You can combine an antibody with an aptamer. You can combine a MIP with an aptamer. You can combine a MIP with a redox active polymer. You can combine MXenes-based scaffolds with MIPs or enzymes. All right. And then, sorry, the fonts are very small here. I gave you some examples, right? So please make sure, if you're really interested in dual receptors, that you click on these DOIs. You've got DOIs on this particular screen. You've got the slides have already been shared. The slides have already been shared. Please click on these. Have a look at these papers. You may not understand everything. Just look at what is the electrode material. You can have your own spreadsheet. And that's a very good exercise. If you really would like to work on biosensing devices and biosensors, go into your area. If you're working on environmental detection, for example, look at the environmental applications. Look at what is the electrode material there. Look at what is the bioreceptor. Look at what is the dynamic range. Look at what modifications have been done. Have your own spreadsheet, right? That will help you have a good grasp on what's going on there. So these papers will also help you in showing you how combining the different bioreceptors together gives you an enhanced sensitivity here. So you can see here going from 3 picograms to 1 picogram per milliliter. This is from 0.09 to 0.012. This one was the best example where from 9,000 you've gone to 3.3 PFU per milliliter. This is actually very, very useful here. Okay, and then you also have this one here, 63 nanometers to 47 nanometers, 1 nanogram per milliliter to 0.5 nanograms per milliliter. Now, picograms per milliliter. I mean, I've shown you examples of dynamic range where we were actually measuring in picograms per milliliter. It's not something which is imagination. You have, your batch has been very lucky that you've been exposed to some latest cutting edge technology, right? Where you've seen examples wherein we've used molecular imprinted polymers to cover a desired dynamic range for biomarkers of Alzheimer's disease, right? You've already seen that. This is an ongoing UK-Japan project. Most of these results will be coming out in papers in the future again. All right, and then I gave you the example of how MXene-based redox systems and molecular imprinted polymers can be used. This is an example wherein you have your MXenes. We saw how these MXenes are made up, and then these MXenes-based redox systems. So again, these MXenes are very useful polymers, which are two-dimensional polymers. I had these examples here. I showed you this. This is the MXene system, which is basically a two-dimensional material. It consists of a transition metal carbide here, and they're basically a class of two-dimensional materials with layered structure, which are composed of transition metal carbides, nitrides, or carbonitrides. Right? This was the MXene that was presented to you. And then we saw how when we combine the MXene-based redox system and molecular imprinted polymers, we managed to get a much more wider dynamic range here. So that was the first example of seeing how you combine two different systems. Now, when I say bioreceptors, you can also have a bioreceptor combined to a nanomaterial system as well. That also could be, I mean, it's not exactly combining two bioreceptors, but it's also an example wherein you have two types of bioreceptors which are basically being combined here, okay? Two types of systems that are being combined here. One is a bioreceptor and the other one is a nanomaterial system here. And then we also looked at a second case study which was on highly porous gold this time as a nanomaterial system, and then combining them with a redox active material, which is your polyphenol red, and polypyridine, which is basically a composite here which is redox active in nature. And then I showed you some nice SCM images. If you want to see more of these kind of nice SCM images here in NIT Rourkela, go and speak to Rishita. She will be able to show you some of her SCM images of the highly porous gold. So there are facilities to do SCM here in NIT Rourkela. So if you want to look at those for curiosity as to what it happens. And then if you remember the example I gave you about these nanomaterial systems, what makes these nanomaterial systems efficient? The fact that they have, well done, they have a very high surface area to volume ratio. And how do you remember that example? By looking at the example from the body, which is that of a microvilli. So if you stretch the microvilli that you have in your intestine and keep on stretching it, and keep on pulling it, you can reach where? To the moon. Well done, you remember the example of moon as well. I'm really glad that you're remembering all these examples. All right, so you have here is the image of this highly porous gold. So when you have a highly porous gold, you have a very high surface area to volume ratio, because within the same volume you've increased the surface area, and when you increase the surface area you increase the number of binding sites, and that's how you can increase the dynamic range as well. Okay, that's that. And then I gave you the example of how this was used for detection of p-tau proteins, and the nice thing here is we're looking at a dynamic range where you have the concentration in the region of picograms per milliliter, right? That's the thing that we need to remember. The dynamic range here is 0 to 21 picograms per milliliter, meaning that you're going from 0 to 21 picograms per milliliter. But then you need to know the sensitivity as well. Of course, the sensitivity is going to be much lower than the dynamic range, and in this particular case, the sensitivity is around 484 femtograms per milliliter. That's the kind of thing. You need to understand the different scales that we use here. You need to understand femtograms is much below picograms. Femtograms is 10 to the power of minus 15, and picograms is minus 12. Nanograms is, there you go. So all of you have a good understanding of that. So because you're going to have, when they're giving a presentation, you need to be able to identify that is this an enhancement or is this a decrease in the sensitivity, right? So that's very, very important there as well. And then finally, I gave you something which I say is hot from the press. Something that has been done as recently as a few days back, some work on monitoring neurofilament light chain for Alzheimer's disease. This is some work that's going on in my lab. This is where the neurofilament light chain comes from. This is a very important biomarker of neuronal degradation. As I mentioned to all of you, neuronal degradation happens in all of us. Once we are born, neuronal degradation starts. But in the beginning, it doesn't actually happen at a much faster rate. But as we start aging, then it happens at a much faster rate, and the concentration of your neurofilament light chain increases much rapidly, right? So you need to be able to capture that, and that's also an indication of dementia, of Alzheimer's disease as well, right? And as I said, people have mentioned this in the literature, that you can predict the onset of Alzheimer's 15 years in advance using ultra-sensitive biosensing devices, right? So you are, as I said, in a fascinating field which has got fascinating applications, right? I mean, that's the whole purpose of being a scientist, right? What do you do science for? Not for prestige. You do science so that you can benefit mankind. And if you can benefit mankind in one way, even if it's a small way, that itself means that your whole objective of performing science or practicing science is achieved here. Okay, and then I gave you this example of nanomips and aptamers. The nanomips, as I said, were the system that's been practiced in my lab, wherein we are using highly porous gold as the nanosystem, and the molecular imprinted polymer is that of polyphenol, is that of phenol red and pyrrole, which we polymerize into polyphenol red and polypyrrole as well. And the aptamers are the ones that have been selected using a particular technique called as SELEX, S-E-L-E-X. That's basically an acronym. That's how you select the aptamers, right? You can go and have a look at that if anybody's interested. People especially who want to be working in genomics need to understand about SELEX systems. So please make sure that you do some research on SELEX as well. SELEX system is used for selecting your own aptamers, and you can select your own aptamers. And these aptamers were selected by our collaborators from Tokyo University of Agriculture and Technology. And this is an example wherein they have got this pink thing. Sorry, the fonts are really small, but can you see the cursor here? So this here is your aptamer, and this is how it binds. It's got these binding sites here. This is what it uses to bind to this large molecule, which is 68 kilodaltons, is your NFL, and it binds to this NFL here as well. Okay, apta sensors for NFL, this is what I'm going to be talking about. So here, this is the results that were obtained by Tokyo University, and the nice thing here is there's one good thing and there's one bad thing, which we all saw. Honestly speaking, you've made my day when you identified what was right and what was wrong here. The nice thing here is selectivity and specificity. Not the best thing here is yes, the range, the dynamic range, right? This is nanomolars. And what are we looking for? Louder. We're looking for picograms per milliliter concentration, right? That's the range we're looking for. On the other hand, so this is NFL. This is some work that has been done. This is done using nanomips here. The nice thing here is you're getting the dynamic range here. This is probably done in PBS, and then this is the next one, which has been done in plasma, and getting a limit of detection of 6.8 plus minus 30 picograms per milliliter here. And then this is when you overlay the two plots, one in plasma and one in PBS. Because PBS doesn't have any interference, right? So you can look at this. Now what do you see here? The nice thing here is you have the desired dynamic range. What is not the nice thing here? Selectivity and specificity here. Okay, so that's what I presented this graph here. So here, look here on the left graph, which is taken from the apta sensors, has got something good. The right graph, which has been taken on nanomips, has got something good. So we would like to make the most of it. So what do? We combine these two together, and we get your apta nanomips and apta nanomip complex here, right? And then when we do the measurements, you can see here, this data is only from PBS. You have managed to, so at the moment it's very hard to say what happens when it comes from plasma. I was hoping I would get some results soon, but they haven't come out yet, but I'm waiting for the plasma results. But I can tell you that for sure that the dynamic range definitely, even with the nanomips, was not giving us the actual dynamic range. We wanted to see something between 0 to 100. So here, at least one good thing that we can see in PBS is that the dynamic range has increased, right? We're getting from 0 to 100 picograms per milliliter. So to summarize, the advantages of dual systems is you can get signal amplification, you can have multi-analyte capabilities, you can improve biocompatibility, you have better reproducibility, and you have modular electro engineering potentials here as well. Okay, let's have a look at the challenges and considerations. Challenges can be how do you combine them together. So fabrication can be a challenge, it can be complex. Interfacial stability, what happens when you bring in two systems together? Are they stable at the interface? How do they react? And then data interpretation, sometimes using machine learning integrated systems, data interpretation becomes hard. And sometimes when you talk about aptamers, then you're bringing in gold as well. You're looking for gold substrates. I'm talking about thin layers, but again, it's slightly, sometimes the material cost can be high as well, right? So that was all that we learned as part of biosensing technologies. That was all that we learned as part of electrochemical biosensors. And then the message I gave you was, we can translate all our biosensing technologies, all the protocols that we develop as part of developing point of care devices, point of care biosensors towards wearable biosensors. So you can use the same. So yesterday, what did you do? You took a PCB electrode which had platinum, you functionalized it with glucose oxidase, and you got a dose response curve, right? If you were to develop a wearable sensor, you would change the platform. Instead of a screen printed electrode, you would have what would you have for epidermal sensors, epidermal wearable ones? Planar electrodes, right? Because you're going to be looking for epidermal, what is the sample here? Sweat, right? Sweat, you don't need to break the skin, right? So what do you need? You need something more like a ring, something that can wrap around your finger, wrap around your wrist, wrap around your arm, right? So you can basically have sheets you can put a layer of planar gold on top of it, and there's ways in which you can do that. There's sputtering methods. I think you can have a chat if you want, if anybody's interested in making those kind of devices. I think the facilities are there in NIT. We have some sputtering facilities. We can get them gold coated, we can get them platinum coated, we can even do iridium oxide and things like that, so there are all those facilities here. So you can actually use this place as a base if anybody wants to pursue biosensors. If anybody wants to do a PhD, you can definitely apply to Dr. Prasoon, Dr. Khalid, and myself as well to see if there are any PhD positions, and we can see exactly, based on the experience, if you want to do a small project or things like that, and get you trained as well if you need that. Okay, so now we move towards wearable devices. Okay, in wearable devices, one of the things we saw that there are different types of biological sample matrices that will allow you to have wearable devices. You can have tears, you can have saliva, you can have sweat, or you can have skin interstitial fluid. Yeah, we focus mainly on epidermal and the subdermal devices. Now, just to let you know, subdermal devices, some people also call them as intradermal devices, right? For example, we call them, we as engineers, as bioengineers, call them as subdermal devices. The animal scientists, the veterinary scientists, they like to call it more as intradermal devices. It's fine as long as there's the term derma in there, wherein basically to show that you're measuring the subdermal plan. Okay, having a look at the next part, which is ISF versus sweat for biosensing. We had a look at this particular slide and we saw how sweat is easy to collect. However, the problem is it's variable and it's contamination prone. Everybody agrees with me on that? And then we looked at skin interstitial fluid. We did agree that skin is the largest organ in the body, and we have a lot more skin interstitial fluid than we have blood in our body. We have 12.5 liters of skin interstitial fluid, while we only have 5 liters of blood. All right, so that's the skin interstitial fluid. Skin interstitial fluid is preferred for subdermal microneedle sensing platforms, right? And then we also, after that, we started looking at the epidermal wearable devices. What are these epidermal wearable devices? I'll show you some examples there. We'll go over those examples one more time. The epidermal wearable devices are basically thin, flexible, conformal devices which can be worn on the skin, as you can see on the screen. They detect sweat-based biomarkers, mainly glucose, lactate, and cortisol. I think we did identify one biomarker which we thought was going to be a very useful one with with epidermal devices, with epidermal wearables. Does anybody remember that? Louder. Well done. Urea. Yeah, we saw that urea in a colorimetric manner is very, very useful to use sweat there, right? And then we also looked at how these epidermal wearables can be used for non-invasive, and they're suitable for wellness tracking as well. Okay, so we move on to the next. We move on to the next thing, wherein basically we have some examples here. We look at the epidermal wearable devices. You can see here, this is one where you use a paper strip here. You have this paper strip wherein this is used for sensing glucose from sweat, and it's combined with monitoring other vital signs, other physical sensors that you have, heart rate, blood oxygen levels, and physical activity as well. This is the epidermal wearable devices. Next thing that you have, example we have is that of a multi-layered epidermal device. Does anybody remember this? This is where you have the electronics on the top, and then you have a microfluidic network so that you can bring in your reagents, you can distribute it, and then it goes down. This is your layer which has got your reference working in and counter electrode, and then this is your adhesion layer which sticks to the body here, right? And this is where, what are these? What kind of sensors are, what kind of bioreceptors are these? When you see a hairpin-like structure, what does it mean? Where do you find hairpin-like structures? In which bioreceptor do you find them? RNA, well done. What are RNA-based bioreceptors called? DNA and RNA-based bioreceptors called? APTA sensors. So in the case of APTA sensors, you get a hairpin-like structure because you have complementary bases towards the end, and they can form hydrogen bonds. So they go up, and there's a loop. They have a loop-like structure there, right? That's how you get these. All right, and the round structure, the rounded part or the spherical part is what we call as the active part, and you normally have a electrochemical tag there. In this case, it's a methylene blue tag which is attached to the top of this, and then you can see this was then used for the detection of cortisol during physical exercise. That's a hormone which is a stress hormone produced during exercise. So that was another example I showed you, and then there was another example which is very useful for female health, female reproductive health. This is where you measure estradiol, which is a very important hormone, right? Why do we need to monitor hormones using continuous monitoring devices? Well done. I think next time you should be here. Excellent, guys. Well done, very well done, because hormones are released in a pulsatile manner, right? So, when we talk about continuous monitoring, when you talk about epidermal devices, or we talk about subdermal wearable devices, why do we need wearable devices? Because wearable devices are mostly required in places where you require continuous monitoring, right? There's this example of measuring glucose using a continuous glucose monitoring sensor where you get a whole profile, and there's this example of glucose being measured using a glucometer where you're getting point measurements. When you do point measurements, there's every chance that you may miss out on the low and the high excursions. You do not get any idea on the direction, the duration, and the magnitude of changes of the blood glucose concentration, right? So you need something which is more continuous. Now, as you mentioned, these are normally produced in a pulsatile manner, right? The hormones are released as pulses. The fertility is decided by the pulses. It's not decided by point measurements. Therefore, if somebody goes to a gynecologist or somebody who works in a clinic or a pathology lab and says, I'm going to take a blood measurement to give you an idea about your estradiol concentration, that's a wrong way of doing things. Maybe that's the only technology that's available. But in principle, if you want an infertility, to address the whole issue of fertility, you need to be able to measure it in a pulsatile manner. Because that pulsatile manner is actually telling you that the body is producing it in a desired way. Because this whole thing is a mix of different hormones being interplayed at the same time. While one of the hormone goes up, the other hormone goes down. That's how it normally works. Epidermal. So this is an example, a very nice example, which was published by Ye et al. In Springer Nature, and this is where they show that how they do the real-time determination of estradiol from sweat, right? Good example, but I think the problem here is you can measure it, but the problem is sweat a reliable sample matrix? I'm going to leave that up to you. If this paper has been published in Nature, clearly there's some value to it as well. And then finally coming to the example that is Malin's favorite example of urea that you have here. You can see here how this particular, you have a variable colorimetric sweat sensor here, which is used for detection of kidney function here. And this was published by an Indian student who was in the US, wherein he's used these for multi-channel colorimetric determination of urea as well. Other examples, this is another one which is, and now after this is an example where you can start integrating the important bits here. This is where, rather than using an external battery, you're using biofuel cells, which are powered by the lactate in the body, right? These kind of things. So this is an example of the epidermal wearable devices as well. And after this, I went straight into the subdermal wearable devices, wherein I gave you examples of how skin, I think I should be having, I missed a slide, which should have been on skin. But I think if you remember, I showed you how, or I might have showed you right at the start of the epidermal devices, of how the skin the microanatomy of the skin, right? So you can tap into the skin interstitial fluid in a minimally invasive manner using microneedle arrays. And to give examples of all the translational research done, I gave you examples of some of my own work. And this was the first human pilot studies that was just done in June 2013, as you can see here. And this is where we had microneedle arrays, which were made up of SU8, and we were using an IV cannula, and we were looking at two important parameters. What are the two important parameters that you look in human pilot studies? What is the first thing that you need to ascertain? Well done, tolerability. And what is the second thing? Specificity. What else? Specificity is normally ascertained during your in vitro studies, right? When you're doing the in vitro studies, you try to get the desired dynamic range. Sorry? That it comes in tolerability. What is the next thing you look at? You look at the performance of the devices, right? So the two important things in clinical studies is tolerability and performance. How do you look at the performance of glucose sensors? Well done. Who said that? Can you say it loudly? Well, okay, Clark grid error. Well done. Clark grid error. And if you have lactate sensors, then BA plots. It stands for bland output plots. Okay, you can research all of these things, and these days, you know, life has given you things on your phone, right? Use your phone more for doing research and looking for things rather than Instagram and all those Facebook kind of things. You know, you have your whole life for the rest of the activities. This is the time when you can concentrate your knowledge and prepare yourself in race for further opportunities, right? And when the opportunity comes, grab it. There is nothing called as good luck. There's nothing called as luck. Luck is when your preparation meets the right opportunity, right? So you will go through multiple opportunities, but only when your preparation has matched the opportunity, that's when you consider to be lucky. That's when you get things in your hand. So don't rely on luck. It's all about your preparation and the opportunity. Okay, coming on to the next part, which is on tolerability of microneedles. So as I said, the first important thing we see look in is tolerability of microneedle arrays, right? So if you look at tolerability of microneedle arrays, the first thing we normally do it by two ways. One is when we use this microneedle arrays or the subdermal devices, the first thing we do is initially what we do is we look at how early do the scars disappear, right? If the scars might disappear in 2 to 3 hours, they may disappear later on. Is there any signs of inflammation? And then after that, when the whole study is completed, we then do something called as a visual assessment method. So for example, if I've done a study on Professor Prasoon here, I would at the end of the study say, okay, can you give me a visual assessment score? What score would you give to the microneedle array? What was your experience been having this for 24 hours in comparison to the implantation of the IV cannula and the comfort of wearing it compared to a different CGM sensor that has been used? So that's where you have this kind of a visual. This graph that you see here is basically a visual assessment, but that is done at the end of the study. But we also look for at the same time we also look for other things such as the scar marks disappearing as well. Okay, and then finally, the other thing that is very important that also kind of evolved during the first study was the instrumentation, right? You have to look at comfort as well, right? You're going to get a very bad score if I put Dr. Prasoon sitting here on a chair for 24 hours without giving him the opportunity to move around. He wouldn't like it, right? This was what was happening here in stage 1, wherein our clinicians who had agreed for the studies, although it was initially our phase 1 was only for 8 hours. But still, you need your natural breaks in between. It definitely is inconvenient there. And then you move from there to the bag. Now this bag was used for 24 hours. Not everybody likes to sleep with a bag, yeah? I mean, we all like to sleep as freely as possible. And then from there, something which was done in home settings, therefore we started using a PCB based potentiostat, right? So use that for studies. Okay, so and then these are some results, and when you plot these results on a Clark grid error, and you can see the points. So what is the main objective here? When do you say your device is working well? When points are in zones A and B, right? That's exactly when you know things are working well. But ideally, what would you like to happen? What is the best thing to happen? Everything is along this line here in the middle. Everything is in zone A, nicely controlled, because this Clark grid error is actually telling you the difference between your sensor readings versus what are the readings that you're getting from a gold standard here. Okay, That was what we did in this thing. And then I gave you examples of how we then moved from, so the first set of studies were done in steady state conditions, right? Wherein a person was, as you saw, person was sitting and relaxing, everything done in a clinical research facility, sitting under an AC, normal temperature, nothing happening, no sweating, no movement, no artifacts during movement, nothing like that, right? The second set of studies, which were done as part of a Ministry of Defense funded project, and in that particular studies, we did the studies, we did simultaneous. Now this time there were two challenges. Number one, not just monitoring glucose, simultaneous monitoring glucose and lactate, not in steady state conditions, not in resting conditions, but in ambulatory conditions, right? This is where we did these studies and we got some good results. And what we found out is, so you could see that we were measuring glucose while the person was lecturing and also running and then we were also measuring lactate during exercise as well. And then we saw that when we take the whole thing and we bring in the AI tools here, we get much better results, which can be seen from this data here. Okay, so what you have here is basically you have your green plots. You can see there's a good correlation. We are trying to look at, as I mentioned, with your sensors, what you're measuring is the current, right? You got to then correlate it to the readings to get your predicted values. So we were using AI tools there and we were getting good Clark grid errors for the glucose sensors and we're getting a good Bland Altman plot for the lactate sensors, right? This was all clear to everybody? So from here, after having done human studies with our subdermal wearable devices, it was then also time to expand into other areas. And one of the areas that we wanted to expand was based on a trip that was made to a farm in Wales. And in this farm, we saw I took these pictures from this farm in Aberystwyth, and you can see here, these animals have got devices which are highly invasive. These are intraruminal devices. These devices have been fit in animals just to monitor the effects of changing their diet. And why is the diet being changed? Sorry, louder, I can't. Do you want to give the microphone here in the front? I can hear you. I can't. Yes, there's this in the front. Did you want to say? Yeah. High GI food. Why high GI food? Why are we trying to change the diet of our poor animals? Are we trying to make them fit? Do we want calorie deficit in animals so that they become fitter? Well, the problem is that the poor animals have been blamed for the greenhouse gas emissions, right? 16% of the greenhouse gases are blamed on these poor animals. So human beings, as selfish as we are, what we do, we change their diet because we have to cut down on the numbers, right? And again, this is also give an example wherein you have link between two different problems that we are talking about. One is the environmental problem of greenhouse emissions and the other one is your biosensors. Can you see how biosensors, although is being used for monitoring of the well-being of the cows, is also in a way helping you address another problem, which is environmental emissions, right? Okay, and then when I looked at the farm, it's not as if these guys don't have point of care devices. It's not as if they don't have any biosensors at all. There are biosensors being used. You have this nice device here, these nice strips which are used for monitoring ketones in cows, right? But the problem you have there is the way you sample these animals, right? As I mentioned, the animals have, you cannot get any capillary blood from them. Can you get capillary blood from animals? Can you ask them to show their paws and can you extract any blood? No, you can't, right? So basically, we started looking into animals and we thought that the animals would be a good candidate, especially high value animals. Some animals are considered to be high value. You can have your continuous monitoring sensors, which are low cost devices, fit in a cow. You can have them fit in a horse, which is a high value. I don't know how many people have heard about horse racing. That's a big industry, right? Especially even in India, I think places close to Pune, they have this horse racing farms where they breed horses for racing, right? It's a big thing, and it's very important to maintain their well-being. It's like preparing the horses for the races. So it's very important that the animal is not under stress. Before you make the animal run, the animal is not going to come and tell you that I'm tired, right? You have to measure the lactate in the animal. You have to measure the cortisol in the animal. You have to measure the ketone in the animal, right? So in animals, continuous monitoring becomes much more important, right? In the animals, it's much more important to monitor their well-being, right? Okay, so the problems we have when we look at animals is number one, they have a thicker skin, right? So it's very hard to actually, so you need sturdier microneedle arrays. They have a different metabolite range, right? They have much lower physiological range. So if for us it's 4 to 8 millimoles, for them it's somewhere around 2 to 4 millimoles. So your sensors have to be more accurate in the lower side of the dynamic range, right? So that's the other thing. They have a different metabolite range. It's difficult to do reference sampling. As I mentioned, you can't get capillary blood out of the animals. The only way you can get blood out of the animals is from their tail. Yeah, you have to puncture the tail to get some venous blood. Again, if you have to do this every half an hour or every 15 minutes, would the animal like it? Exactly. And then there's also the tolerability assessment, and then there are also bioinstrumentation restraints. I showed you some pictures of some studies that were done in a farm, not in a farm, but at a place where they brought in freshly dead animals. Horrible conditions. Even the farm where you have very happy, lively animals and the calves and everything, they look all very nice. It's contaminated with the animal feces, with cow dung and everything, right? It's very hard. As I said, I gave you the example. My preferred anatomical location to do studies in milking cows would have been the udder, but the udder is so close to the floor that you would definitely have the udder contaminated. So how do you save your sensor from? That's a challenge. I will still manage to do it by whatever I have to do it, but that's a big challenge. So bioinstrumentation constraints is a big problem when it comes to animal studies. And then I showed you the design that we used. I showed you how we use these acupuncture needles, which are much more sturdier and commercially available. We used an applicator, and we used the PCB-based potentiostat here as well. And then coming to the insertion studies, these are some nice pictures to show you what happens to our needles pre and post insertion. As you can see, they're still mechanically robust. There's the tip still looks to be intact, no loss. Everything looks to be perfectly fine. And then finally, I showed you this nice video of this. This is a beef cow. It's not a milking cow. And then I showed you how the devices were. You can see here from here, nicely inserted. But here you can see the hair had to be shaved off, just to make sure that there is one less variable, and then you take off the applicator. Now we are designing applicators, and we're designing devices that can be inserted onto the animal without having to shave them off. Yeah. Okay. And then we also looked at the challenges in future directions. One of the biggest problems that we have and will have with these sensors, with these subdermal sensors, is the sensor drift. We have the drift problem there as well. So yesterday, if you remember, when you were doing chronoamperometry, you would have seen, or if you do a chronoamperometry with 0 millimole solution, you can see that you normally have a sensor, it drops down, it still keeps on drifting for a long time. You have to stabilize it. It has to be stabilized over a certain period of time. Normally, when you're doing these microneedle arrays, you have a much larger surface area. So when you have the sensor, you leave them to stabilize for 15 minutes till they come down all the way. Any idea, anybody? Why do we have this drift? Okay, there are two reasons for this drift. One is a normal drift, a natural drift that you would always see. The other reason is, anybody? I'll get Rishita to answer this later on, but I'll give others the chance first. So what are we polymerizing with? What are we polymerizing on the sensor surface? Phenol. Any idea now? Have you got a hint now? What can happen there? What is phenol being polymerized into? Polyphenol. So what's happening there? If the reaction doesn't complete? Louder. It forms an insulating layer. That's fine. But where is the second part of the drift coming from? Why do we need stabilization? Well done. I didn't know that you were paying so much attention on the day. So we have unreacted phenols as well, right? The unreacted phenols are also something which can cause a drift, right? Because unreacted phenols are conducting. Phenol is insulating, right? Just to make you understand the fundamentals, the molecular mechanisms, as to where the contributions can come from, right? And then the other problem we have is limited long-term data and regulatory hurdles. I showed you the example of the human studies we did as part of the DASA project. What is the big problem there? We only had ethical approvals to do studies between 8 a.m. to 5 p.m., right? Because and we were not allowed to do any studies in the night. That kills the whole objective of the studies. We get data, that's fine, and I also showed you data from one of the previous studies wherein I showed you that we know that after 24 hours, what sets in? Even before 24 hours, something sets in, which is the body's response, which is defined as the body's response. What is that called? Starts with a B. Biofouling. Well done. Yeah, so the biofouling sets in within the first 24 hours, and I showed you the results where I had those pyramids there showing you the biofouling effects. So clearly there are regulatory hurdles. There's limited long-term data. If you look at publications, this is something we've discussed so much over the last few days, is that people are publishing papers, but those papers only have got data for we've got a fantastic sensor here which can do multiplex sensing, blah, 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 but those are only done in participants over a range of 2 to 6 hours, 2 to 4 hours, and they're publishing in Nature. That's the problem we have. We don't have data wherein you can, like the animal studies that I've shown you. This is data that has been obtained over 24 hours of studies. I didn't go into much details because I thought there was going to be a lot of information in here already, and you already have seen the graphs, and you know what the important thing is to see in a Clark grid error, right? So next time when you see a Clark grid error, what are you going to be looking at? Zones A and B. And when you look at Bland-Altman plot, what are you going to look at? There's a straight line in the middle, and there are 95. How many points are between the 95% acceptance limit, right? If you've already understood how to look this, good news for you is you know how to read a paper on wearable sensors. You are now ready. So now there won't be a barrier. Earlier what happens is when we don't understand anything, we are reluctant to read that particular paper. Now you understand wearable technologies. Now you understand biosensing technologies. Now you understand point of care biosensors. Now it's not going to be a big challenge. Even for those of you who are working on physical sensors, how do you look at the data? How do you look at the performance of the devices? You now know how to measure that. You now know how to look at tolerability is going to be common, right? If the Apple Watch, before they made these Apple Watch, do you think they would have not tested these on other people? We saw the example of GlucoWatch that. And we know that the reason GlucoWatch failed was because it was not the technology that failed it, it was the adhesive that was used, which was giving rashes to people. They changed the rashes, and then suddenly they've started getting good results. And then hopefully we should have more good results based on some of the new results that have come out of NIT Rourkela as well. And the final challenge that we have with subdermal listening is getting in the AI, multiplexed sensing, real-time analytics. I did mention that when we were doing the studies in humans, the sensor was designed so that we would have a glucose sensor and a lactate sensor. However, the bioinstrumentation that was available, although it claimed to be a multi-channel potentiostat with a multiplexer, wouldn't allow us to measure more than one channel. So we had to attach two devices with two potentiostats. So imagine both the hands with two devices measuring glucose and lactate, and then asking this person to then run on a treadmill and cycle and do things like that, right? It's really hard. It's difficult. We had about 15 participants, but before the participants, I tried them on myself, and I try to do the same thing. As scientists, you have to be conscientious, especially when you're doing the animal studies. You have to feel the pain yourself first before you propose it to be inserted in other species, whether it's animals or humans. You got to be very, very conscientious about that. And ethics, integrity, these are all very important part of science. So the next steps we think will be bringing in the AI integration. We think we will address the challenge of multiplex sensing, and then we also think that we should be able to have something which will give real-time analytics. At the moment, all the results that we are showing you is all data that is being processed, and then we are getting the data results there. We would like something to be similar to the CGM sensors that we have. Even the CGM sensors, it's processing over time, and then it's throwing a data value every 15 minutes. We would like to have something which is a bit more faster, an electronic architecture, which I think hopefully will be designed with Professor Khalid's group, wherein we have real-time values. Oh, your value is this much. But then there are pros and cons there, right? As I said to you, the reason the continuous glucose monitoring sensors had poor acceptance at the start was because of the fact that they are devices that are implanted deep into the body, right? When your technology is fighting the body is definitely going to win, right? So how do you address the problem of biofouling? You can address the problem of biofouling, number one, by bringing in a membrane that is perm selective, that is only selective to the glucose molecules and does not allow the proteins that are being produced by the body to cover the sensor surface. That's number one, right? Over time, it will go and cover the membrane, but none of them will reach the sensor surface. And what will happen, even if it covers the membrane, the amount of glucose that will diffuse through the membrane onto the sensor surface will go down. So there's no way you can fight it, right? The body is definitely going to win. The other thing you can do is you can do a proper study using machine learning tools or deep learning tools, bring in your AI models and then predict values. But again, those values would be predicted values. Those will not be the absolute accurate values. You will always see a difference in your blood glucose reading done between two devices in the body because they use two different AI models, right? I think that was all about the presentation I had for you. That was all that was presented. What I'll do is I'll give you 5 minutes and I'll open the floor for any questions. This is my email address. Some of you have already sent me LinkedIn requests to join to this thing. I have already approved some of them I haven't. I just want to be sure because I keep on getting a lot of requests, especially since I've been to the World Congress on Biosensors as well. I just want to make sure that it is relevant to your interest as well. And I'll be more than happy to connect via LinkedIn, and I'll be more than happy to share my research insights. Most of it normally comes up on LinkedIn every time we have a presentation at the conference or we have a paper publication. So if you are already attached to or connected on the LinkedIn site, that's a good thing. I'm not on Instagram. I don't do Instagram. That's for you young generation. I'm too old for Instagram. Facebook is more personal, so I don't give my Facebook to anybody. But yeah, I think LinkedIn is a good site, a good platform to connect and to keep in touch.
Hello. Am I audible? Yeah.
Yeah.
So, Sanjiv, just one of in the slides you talked about the MIP based sensors.
Yes.
So, the fabrication process, as you mentioned, there are certain challenges as well.
Yeah.
So, do you think that currently the state of art says that this MIP based sensors can be a scalable way of making those sensors which anybody can use it, or still there is a lot of the hurdles which need to be overcome? What's your opinion?
Well, the slides that I showed you, I mean, for the lab scale, for the research purposes, that's perfect because you're talking about electropolymerization. Now, it's a good question you've asked. All right. This is a nice question Dr. Prasoon has asked, and I want the answer to come from the audience, right? When you talk about medical devices, what is the most important thing that you need? Most important thing to make it acceptable globally?
Standardizations.
Standardization. Before that, even standardization. Sorry? Biocompatibility, what else? What is the most important thing when you talk about medical devices? When is a device most easily acceptable? Louder.
Safe.
Cost. So to make it cost effective, what do you need for making it cost effective? Well done. Sustainable material. What else? How do you make it out of? So what do you need for that to make them for manufacturing? Scalable technologies. Right? So if you have something that is, so at the moment in the lab, we are doing electropolymerization. The question to you is, and then that's where you're going to answer Dr. Prasoon, is electropolymerization a scalable technology? I want you to think about it. Is it or is it not? I think it's not. Why? Because imagine, how many engineers do we have in the room? How many engineers? All right, engineers, come on, help, come and save me. When you talk about, if you were to make electropolymerized, if you were to have molecular imprinted polymers, I mean basically you need polymerization. Is electropolymerization the way forward when it comes to scalable technologies, or would you prefer something else? Have you heard about robotic technology, robotic arm, automatic dispensing? You need something like that. Can you do polymerization? Can you do electropolymerization with a robotic arm? You can, but it's going to be very time consuming. It's going to be very laborious. So you need something where you do the polymerization without applying an electric field. That's your answer.
Okay, one more question. So in the examples which you showed, phenols were used for polymerization and making the polyphenols. So there could be the other type of the also materials which could be used.
There's a lot more. I mean, there is actually a review article on molecular imprinted polymers, which I think everybody should read. There are many of these molecular imprinted polymers because there is a list of materials.
Okay.
And you can, as I said, excuse me, there is a list of materials, and you can actually use those materials. You can combine those materials. You can combine the good things of different materials. Something would be better conductive, something would be better redox properties, something will work at a different potential, something will work at a much lower potential. There's a lot more different materials you can combine, and there's a list, organic compounds. I mean, any chemical engineers in the room? Can you give me a rough idea as to how many organic compounds you have? A lot, and most of a lot of phenolic compounds, a lot of these, the rings which have aromatic ring compounds or cyclic ring compounds, a lot of them have got redox properties, and you can exploit them. But I like the idea of sustainable materials. Well done.
Any other question, guys? So the floor is open for questioning.
Are we all ready for the quiz then?
I think they are ready for quiz.
All right. If there's no more questions, I mean, looking at your responses, me personally, I'm mightily pleased. Very, very pleased. Thank you so much, all of you, for paying attention, attending the lectures regularly, and answering. As I say, my students inspire me to. I mean, it's everywhere, whether it's in the UK or whether it's in India. India even so a lot more. It inspires me, to kind of give my best. I cannot explain to you. I'm not in the best of. I've overdosed myself with paracetamol and cetrazine, and to stand here so that I can give you a final talk. But I'm really, really happy that that all of you have been very responsive. Thank you so much for that.
Thank you, Professor Sanjiv.
Let's wait for Professor Khalid, because I think he also wants to take the quiz. Number 2, what is a key benefit of using molecular imprinted polymers in biosensors? What is a key benefit of using molecular imprinted polymers in biosensors? Your options are require complex chain logistics, limited stability in harsh conditions, high specificity and low cost, or only suitable for enzymatic targets. Your time starts now. Read the options carefully. What is a key benefit of using molecular imprinted polymers in biosensors? Everybody done? Does anybody need more time? You're all doing it within less than 30 seconds, which is making it harder for me. Okay, I'll get rid of the timer. Let's move to question number 3. What is one major advantage of dual receptor biosensing systems over single bioreceptor designs? What is one major advantage of dual receptor biosensing systems over single bioreceptor designs? A, simplified fabrication. B, reduced biocompatibility. C, enhanced signal amplification. And D, lower selectivity. Done? Shall we move to the next question? Yeah? Question number 4 on your screen. In the MXene MIP redox sensor, what role does polyaniline play? Is it a reference material? Is it an enzyme mimic? Is it a conductive polymer scaffold? Or is it an optical label? Is it a reference material? Is it an enzyme mimic? Is it a conductive polymer scaffold? Or is it an optical label? If you remember the case study we did on MXene with polyaniline, are we all ready to move to the next question? Question number 5 on your screen. What is a reported challenge in animal wearable biosensing? I mean, this is one of the answers that people from this audience would know because you had a lecture on we had a case study on animal wearable biosensing. A, excessive sweat rates. B, unreliable wireless transmission. C, thicker skin and metabolite variability. D, short battery life. What is a reported challenge there? What was the reported challenge in the animal wearable biosensing, that case study that we did? Was it the excessive sweat rates? Was it unreliable wireless transmission? Was it thicker skin and metabolite variability? Or was it short battery life? Everybody, are we happy to move to the next one? Anybody who needs some more time? No? All right, let's move to our next question, which is question number 6. In the first human pilot study using microneedles, which results supported tolerability? In the first human pilot study using microneedles, which results supported tolerability? Was it the continuous bleeding at the insertion site? Was it persistent inflammation at 48 hours? Or was it the disappearance of marks within 1 to 3 hours? Or was it dermatitis reaction in 60% of the subjects? Which result supported tolerability? Dermatitis is inflammation of the skin. It's an adverse reaction. So which result supported the tolerability of the device? Everybody done? Shall we move to the next question? Yeah, Tushar? Done? Question number 7. Which of the following is not a typical material used in temporary tattoo electrodes? We saw temporary tattoo electrodes when we were doing epidermal wearables. So which of the following materials is not a typical material that you would use in a temporary tattoo electrode? P.PSS, stainless steel, gold nanoparticles, and stretchable polyamide. P.PSS, which is basically a polystyrene sulfonate, stainless steel, gold nanoparticles, stretchable polyamide. Everybody happy to move to the next question? Last 3 questions left. How are we doing? Everybody feeling confident? Good. Shall we move to the last 3 questions there? Now this might be something which you may not remember the exact figure, but the answer is in the question here. What was the detection limit? I'm not asking about the dynamic range. What was the detection limit achieved for the p-tau 181 using highly porous gold, polyphenol red, and polypyrrole system? What was the detection limit? 5 nanograms per milliliter, 484 femtograms per milliliter, 21 picograms per milliliter, or 3050 picograms per milliliter. If you want, I can remind you that the dynamic range is 0 to 100 picograms per milliliter. All right. Everybody happy? Move to the next one. Question number 9 on your screen. What is a primary challenge in a long-term epidermal wearable usage? What is a primary challenge in a long-term epidermal wearable usage? High user dropout, sensor biofouling and signal drift, too much glucose in sweat, or poor adhesion on the face only? What is a primary challenge in long-term epidermal wearable usage? High user dropout, sensor biofouling and signal drift, too much glucose in sweat, poor adhesion on the face only. Thank you, sir. कैमरा तो नहीं चल रहा मेरी तरफ तो नहीं. Everybody okay to move to the last question? Yeah. Question number 10. What type of analysis was used to assess the accuracy in the subdermal glucose monitoring human trial? What type of analysis was used to assess accuracy in the subdermal glucose monitoring human trial? Did you use the Bland-Altman? Again, this is glucose here. Did you use the Bland-Altman plot? Did you use the ROC curve? Did you use the Clark error grid, or did you use a fast Fourier transform? What type of analysis was used to assess accuracy in subdermal glucose monitoring human trial? Everybody done with it? Do you want me to run over all the questions one more time, or did anybody miss any questions? Do you want me to repeat it? No? Everybody okay with this? All right. Yeah? Okay. In that case, shall we start looking into the answers? Please mark your own scripts. Don't mark anybody else's scripts. Yeah? And don't look into the other person's scripts. Let them, as I said, you versus you. All right? Mark your own answers. All right. Question number 1. On your screen, which biofluid is considered most stable and reflective of blood chemistry for diagnostic purposes in wearables biosensing? The answer is skin interstitial fluid. How many of you didn't get it? Did not get it? Listen to the question first. So everybody got it. Well done. You've raised the bar up now, so I don't expect anything less than this. Question number 2. What is a key benefit of using molecular imprinted polymers in biosensors? The answer is high specificity and low costs. Did anybody get it wrong? Yes, what did you get? You got what? Only suitable for enzymatic targets. That was a guess, right? I mean, I would have expected things to go wrong because of the discussion we had with Professor Prasoon, because we spoke about costs there, and electropolymerized polyphenol would not be cost effective, but if you can polymerize it using robotic handling systems, it becomes very cost effective. Okay? Question number 3. What is the one major advantage of dual receptor biosensing systems over signal bioreceptor designs? How many of you did not get it right? You didn't get it right? What did you get? You got it right. So listen to the question. Did you also get it wrong? Did you get it wrong? What did you get? Simplified fabrication. That is close. That would have been the second answer. So you can mark it right. That's actually because you remember we were looking at the challenges, and sometimes getting them together interfacial. But I mean, I think if you ask about the one major advantage, that would have been something less than a major, but it's still an advantage. So I'll allow you to mark it right. Question number 4. In the amexine MIP redox sensor, what role does polyaniline play? How many of you got this right? Majority of you got it right. Well done. Well, it's basically a conductive polymer scaffold, right, which basically helps with the amexine system for the electron transport. Okay. Next one. Question number 5. What is a reported challenge in animal wearable biosensing? Sorry? How many of you did not get it right? Well done. Because this, if you get this answer right, that actually means is that you're ready for the animal studies on a farm side. Simple as that. No, seriously, I mean it, because it takes, people work in the lab doing in vitro optimization and things like that for 2 to 3 years. I've had this project running since 2019, 2020, after the pandemic. And it's only now we've started seeing. So 5 years to realize this whole problem. So you're all gone wiser by 5 years. Next question. In the human pilot study using microneedles, which results supported tolerability? Yeah, disappearance of marks within the within 1 to 3 hours, because the rest of them appear to be not supporting tolerability. Okay. Question number 7. Which of the following is not a typical material used in temporary tattoo electrodes? Why? Mechanical properties, it's not very flexible. Electrochemical properties, is stainless steel conductive? But not as good as gold and other materials. You have to electroplate them, because we buy, for the, if I remember clearly mentioning, when we buy the acupuncture needles, they come made up of stainless steel, because that gives you the required mechanical stability. And they're sturdier, right? For animal studies, they are the ones that we use, the acupuncture ones. But when it comes to functionalizing them, they give us very poor electrochemical behavior. It's very hard. So we have to electroplate them. You're right, they're conducting, therefore we can electroplate them. You cannot electroplate anything that's inert. So we electroplate them with another metal layer, and then we make our sensing devices. Question number 8 now. In the lecture 5, what was the detection limit achieved for p-tau 181 using the highly porous gold polyphenol red polypyrrole system? Again, the question here is limit of detection. Limit of detection is always a low value there. How many of you got this wrong? Oh my God. My star students got it wrong. So you guys will have an extra lesson today. The answer is 484 femtograms per milliliter. What is a primary challenge in long-term epidermal wearable usage? How many of you got this right? It's sensor biofouling and signal drift. Well done. Moving to next question, question number 10. What type of analysis was used to assess accuracy in the subdermal glucose monitoring human trial? The answer is, and if the question was about lactate, well done, blind Alpin plots. I think that brings us to the end of this part of the quiz, and we'll soon try to organize your next part of the quiz or next part of the exam, which is going to be oral presentations, right? To give you some, how many of you have done oral presentations in the past? Everyone has done oral presentations, so it's nothing to be nervous about, right? Well done. We'll have lectures for him for those lessons. As I said, this is you versus you. So you're not comparing your marks to anybody. And I don't want to know the marks. If you've got anything wrong, is there any questions you have for me where you got it wrong and you think it was closer? Anybody who's got an answer which is, yes, Satya? Which question? Question number 8, you were thinking? Sorry, one more time. For question number 8, what did you think?
Sir, I was actually thinking for
Please
B.
Please pay attention here, it will benefit you as well.
Yes, I was thinking actually for B. Then I just got, confusion that it's for lower or higher. That's why I just...
Limit of detection is always, what is your limit of detection? It's mainly the lower one.
Okay. So
unless
that's...
it says LLOC, it's normally the term, if it's limit of detection, then it's the lower one.
Yeah.
If you want to talk about lower and higher, the dynamic range, then you talk about lower limit of quantification, higher limit of quantification. Yes. LLOQ and HLOQ.
HLOQ.
But when it's limit of detection, it's always, what is your limit?
Yes.
How low can you go down? Because that's going to tell you about how sensitive your assay is.
Sensitivity, yes.
That's fine.
So next session we'll do a group discussion.
Yeah.
So what we'll do is in the next session we'll make some groups. You can form your own groups. Then what is the idea?
And then what I'm going to give you a task. So you can form your own group. I'll give you a task. It will be a simple task wherein you will be asked to design an epidermal device or a subdermal device, your choice is yours. You have to come back and say, our group thinks that this is the problem, major problem. You got to come give me some statistics. If you're developing something on the lines of urea sensor, for example, say, oh, urea sensor, urea is a biomarker for acute kidney disorder or something like that, and there are so many people suffering from this particular disorder. It's very important. We need to measure this, and therefore we would like to measure this using, epidermal or subdermal. Why epidermal? Why subdermal? What is your case study for that? And then a proposed design. You can just draw something bare hand, take a picture of it, and stick it onto on your presentation. Or if you want to design, if you have the time, you can sit and do a PowerPoint design. Is that okay?
So just to summarize what Sanjiv just told. So first of all, you have to decide what sensor, which analyte you want to monitor. Okay, first.
Your clinical application. Your application is the important thing.
Yeah.
Why? I mean, if I talk in terms of layman, what is the need?
Yeah.
How is your sensor going to impact that need? Right. Second thing is, what are you thinking about the cost of your sensor? Why? I mean, why are you choosing what are the materials you're going to build your sensor with? Right? Is it going to be a cost-effective sensor? How much do you think? Just a quick calculation of your head. How much will that sensor cost? Right? And then thirdly, why are you going with epidermal and why not subdermal? And why are you going with subdermal and why not epidermal? Right? And do a presentation. So you can have, if you have a group of 5 to 7 people, not everybody has to present. Maybe just do on each point, just present for like 2 minutes. I don't think you should keep 5 minutes presentation followed by 3 minutes of Q&A, and where everybody else will be given the chance to ask questions.
So yeah, that's
so
all.
8 minutes will be fine, and we can have
Okay, ready? So you use your mic, please. Huh? Mic for.
हेलो 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 सेट नहीं ऐसे हाथ में पकड़ लूंगा तुझे भी दूंगा ना हेलो हेलो हेलो
ये रख देना, आपसे कुछ क्वेश्चन करना हो.
Ready, boys? Everybody ready here? So you'll have 5 minutes for presentation and followed by 3 minutes for Q&A. So what's your group called?
Warriors.
Warriors. Warriors. And you only have 3 people?
Yes sir.
All right, that's fine. Sure. You want to make a start? Yeah, go on.
So good afternoon everyone. My name is Aditya Bushola.
Abhishek Kumar Gupta.
My name is Kiran Kumar Bindhani.
So first of all, I want to thank Dr. Sanjiv sir, Dr. Khalid sir and Dr. Prasoon sir for this wonderful, insightful presentations. And after inspired by that, we are came up with a great idea. And actually we are focusing on a epidermal level. So here the idea is a smart ring for glucose monitoring. As we all know, this is the non-invasive smart ring design for continuous diabetes management. And first of all, everyone knows what's the importance, so we are not deep diving into the details. And current methods, we have these sensors we have seen, and the challenges which we are facing currently is that we have to actually penetrate our epidermal skin for the monitoring, and we want to actually replace that. We don't want to penetrate our skin for removing blood, and this method is not for real time monitoring. So we really want to make it continuous monitoring. So we want to make a device which is epidermal, easy to wear, and so that's why we are here for with the ring.
So here is the proposed solution, the smart ring. It's a novel smart ring design which can be fit into the ring finger or any finger, and basically it use the RF and microwave frequencies that penetrate into the skin, and then it will capture the changes in the dielectric constant when the glucose is there in the blood or not, and other metabolites like fats and water and every components of the blood, and accordingly it measures the change, and then after that it will give the data, and then we will process it using machine learning and all. So the technology behind it, I have explained it. So this is the signal processing and machine learning aspect. Over to you, Aditya. You can explain it.
As Abhishek said, that we will be using radio frequencies and microwaves. And the idea of using this is that actually we are not penetrating through the skin, but actually we are giving the signal through our skin. And after that, we are getting the changes in the dielectric constant of the body. That, as we all know, there are so many water molecules, fat and lipids, there everything, but there is also glucose, so, and that is changing in the diabetes. So we are actually countering with that, and we want to measure we want to measure the minimum changes are there, so we can increase our efficiency. So that's why we have used these type of technology. And actually why we have talked about that, how we will be measuring these things. So this compact ring will actually enhance the way of measuring glucose in the body from the previous. So these are the some signal processing step which will be running in the background, and we can actually filter the data to find the minimum changes in the glucose with the help of dielectric. And actually we are changing the way of measuring, so here you can wear the ring anytime. You don't need to remove it while sleeping also. That was the problem we have seen in the presentations. The most recent technology, that is artificial intelligence, we will be incorporating it with our device, and due to that we can actually find the changes in the future will be gonna happen from the data which we will be feeding into our ring. And due to the time restraint, we can tell this.
Then the data after processed will be given to the physician and all, and then he can predict. Thank you so much, sir.
Okay. All right. Can we give a applause for the presentation, please? All right. So, from what I understand, basically you're talking about an epidermal sensor which relies on the dielectric variations of the glucose concentration, and you have the machine learning and the AI to account for any interferences that you may have, and the information will be collected and relayed to a physician, and that's how it's going to work.
Yes.
Right? Can I request a few things, please? Number one, can we please maintain a little bit of quietness when the presentations are going on, because it's a little bit unfair on the people who are presenting, and if possible, please also pay attention to their presentation styles. You can learn a lot of things there as well. All right. I'm going to open the floor for questions. Has anybody got any questions for the group? Yes, please.
In what way the person would be specific to that any other changes the dynamic GIAN structure.
So for that, first of all, we have to collect the clinical data, like CGM sensor, from the CGM sensor, and then comparing it with the RF data, and then process that. First of all, we have to do the clinical things, and then we will implement this for the specificity. Specificity, sir, it will be done by using clinical data. It will be done.
But this whole concept of using dielectric variations,
Yes, sir.
is there enough, is this idea that can fly? Is there enough evidence there? Do you have enough literature review on the use of technology where we are using the dielectric variation to use that?
And then it will penetrate through the skin to the dermis layer, and then the interstitial fluid will be.
So it's not a sweat-based epidermal
No,
sensor, it's
no sweat, no ISF, and no anything.
it's definitely has to be ISF because you're measuring the ISF, but the epidermal ISF,
But not invasive, na?
not in this.
Yes, sir.
Yeah, it's a completely new concept there. Has everybody understood what they're ideating? It's a new concept where you're not using micro needles, but you're still monitoring in variables and you're tapping into the skin.
Yes, sir. So till now there are rings for sleep monitoring and heart rate monitoring, but there is no specific for glucose monitoring.
So if the fundamentals work, if the basics work, then there's no reason why this won't be a big hit.
Yes, sir. Yes.
Anybody has any other questions? So, all right, then. Thank you so much. This is group 1 warriors. We have a second group. It sounds more like the IPL. Yeah? Krishna, organize packets. No. Evidence. Any more variants? Or challenges? Or language? How would you like that? No? What about the generation proteins here? In the front? Not ready yet? We're giving you 5 more minutes and then we'll randomly call up the groups. Now, okay, because we are a little bit pressed for time, we don't want to delay your lunches and then we also have to plan the afternoons. It doesn't have to be too refined, no more. It doesn't have to be too refined, it's just. It doesn't have to be something that makes me afraid, it has to be something that
So as we know that we have a task to design a subdermal wearable sensor. So I have come with an issue that's going on worldwide, that we have also seen a very good disease or a very well established disease, that is iron deficiency anemia, which is being it's getting overloaded over us. So now what is actually this iron deficiency anemia? Now if the deficiency number so the anemia is the deficiency of number of red blood cell or deficiency in total hemoglobin content per unit of the blood. So if the body synthesizes the hemoglobin and this hemoglobin is not getting that much sufficient for the this oxygen transportation in the blood in our body, so this situation can be told as the iron deficiency anemia. So what are the causes here? So the causes here are excessive blood loss or blood destruction by drug therapy, infection or toxins, or defective hemoglobins like thalassemia. That's also a disease. So what are the things, what are the present scenario? Right now we are being cured by taking the iron supplements. So what are the iron supplements? These are the iron 2 drugs. So whenever we take the iron 2 drugs, the problem is, one of the major problem is the Fenton's reaction. This Fenton's reaction, whenever it goes inside our body, it produces the hydroxyl radicals, which are very invasive in nature. So it can also cause the blood cell damages and also some cell these tissue damages. So what else we can do? So this is the target approach, transferrin. What is transferrin? Transferrin is a iron, it's a iron binding protein, which it takes the iron from the cell. Whenever we are taking the drugs or something, it takes the iron from the cell to the from the blood to the cell. So now if we can target this transferrin, we can find out what is the iron supplements are doing with the doing inside the blood or with the cells. Wherever we also have seen that the things, whenever this anemia level is increased, the transferrin level that goes on increased, means it's inversely proportional. So it's like whenever the iron supplements will be there, that the body tries to it regulates or it produces more number of transferrin to put the irons to take the irons inside the cell from the blood. So we can target this whenever we're taking the drugs or something, or we are having anemic. So this transferrin level can increase. So if the transferrin level is increased, then we can also target it. Now, so in the ISF, the transferrin level is 0.2 to 0.5 gram per liter. Now these are the things, and its redox potential is found to be minus 0.4. Whenever it binds to iron, its redox potential changes to 0.5. That means there is no that much difference that we cannot monitor where if it is binding the iron or not. Okay, so wherever there will be transferrin, we can measure it. So this is the transferrin in the human, and we can see there are the heme, which it's the apotransferrin, and whenever it binds the iron, it becomes like this, and these all two things are in similar redox potential, which is a very good benefit for us. So we can measure any situation whenever we are targeting a patient or anyone. So there will be no difference whenever we are taking drugs or not. We can measure the transferrin levels with the time and continuously also, the whole day. Now how can we design the electrode? Now we can design the electrode by modifying the electrode surface by one of the transferrin receptors. There are hepcidin and all. So we can also design the electrode by doing the modification on the electrode surface, which we can also measure by the electrochemical measurements. And as I have shown you that transferrin level increases when the blood iron level decreases. So transferrin level can be measured, and we can also get it from the electrochemical measurements, and this is done. So now the thing is, when I have not made that conclusion slide, but I can tell you that the thing is, we are going to make it for the subdermal devices, because I have shown it that we are measuring through the ISF. So we can do it with a very beneficial idea. Thank you, sir.
Right? I think that's a very nice presentation, especially with all those presentations we have on transferrin. Give you an idea about it. So basically, you're trying to develop a subdermal transferrin sensor.
Yes, sir.
Yeah?
Yeah.
Everybody agrees that you understood that's the message you've got, that we are trying to develop a transferrin. So what's your bioreceptor for the transferrin sensor?
Sir, I think the hepcidin can be.
Sorry?
Hepcidin.
Hepcidin.
Yes.
Okay. Did you mention that in your presentation?
No, sir, I have not mentioned it.
So if you're talking about sensors, because I think I got a feeling that you focused a lot on the mechanisms of transferrin binding and things like that, but this is more meant to be a presentation. I mean, we are not trying to understand the fundamentals behind transferrin binding and things like that. This is where, here's the sensor, this is how we expect it to work, and this is going to be very useful. So if you talk about subdermal, you're talking about how are you going to be accessing the interstitial fluid?
Yes.
How will you access it?
So like with the micro electrodes
So
and
you're
all.
going to use the microneedles there.
Yes.
What are your microneedles going to be made up of?
This
Is this for human or is this for animals?
for human.
For humans. So what are your microneedles going to be made up of?
So maybe gold or platinum,
Gold or platinum will be the coating, but the base material will be
stainless steel.
stainless steel. Alright, so it's not more acupuncture needles. That sounds like a good idea.
Yes.
Fantastic. And you will have that, and then you're going to immobilize hepcidin, did you say?
Yes.
On top of that. Is
Yes.
hepcidin easy to, and you mentioned about something about your dynamic ranges, but did you say 0.2 to 0.5 grams per liter?
Yes.
Which is 0.2 to 0.5 milligrams per milliliter, which is again in some very high concentration. So you should be able to do the measurements. I think we're talking about millimolar concentration here.
Yes, sir.
Or micromolar concentration.
Yes. Micromolar concentration.
Micromolar.
Yes, sir.
That's really good. That's really good information that we need to have. So you can confirm that. So anything you can do with this afterwards.
Yes.
Very good idea. And how many people are affected by anemia?
So worldwide, many people, sir. We
Many
cannot.
people.
Yes, sir.
So clearly, there's a need, there's a big impact there as well that you're working with. I think that's actually a very good idea you have there on your transferring subdermal devices.
Yes.
Anyways, got any questions for this group?
Yeah. So you are measuring the blood but that is the subdermal one. So how can it is correlated with the liquid?
Sir, actually there is a group Kaur et al. They have done with the serum level.
Okay.
So they have done with the serum level because we know that the transferrin level is very high in the serum. So we can also correlate with them. And also there are different proteins which are biomarkers for iron. So
Okay.
we can also correlate with this diagram or these sensitivities with the gold standards using
Okay.
worldwide.
That makes a lot of sense.
Yeah.
But any more questions from the rest of the groups? No, in that case, let's have a round of applause for the black fighters. Okay, and we have to invite the next group. Who's ready? Yeah.
Yeah, you have to really everyone has
So we are presenting Cortisons, a continuous real-time stress monitoring system. But before I present to you what we have thought, I want to share one anecdote that I had last year. So last year, because of some complications, I had to admit myself to ICU for kidney problems. And there with me, there were another 5 patients. The only thing that stuck with me was that for various reasons, all of them were in very much pain. Somebody had a very major COPD attack, few had fevers, and I also there. And being from biomedical background, I had noticed that although we test for different types of markers, there is a continuous heartbeat monitoring is going, pulse monitoring is going, everybody is checking the SpO2. But the fundamental marker that we associate with stress, that is cortisol, nobody is checking that. In fact, this problem is pertinent in certain cases of clinical trial also, as I have heard from people who conduct these clinical trials in India. To be very specific, to the eastern part of India, this is a very big issue. So having that in mind, we have came across this idea that how about we target this cortisol hormone. So as you know that the cases of depression, anxiety, and which leads to death also sometimes, very sadly, is very frequent in India right now. 75% of the adults report of regular stress. And there are many invasive techniques already available. For example, you can take the capillary blood and you can test for cortisol. But having my parents who have diabetes for last 25 years, I have seen my family members and my friends are very reluctant to give any blood samples. It's a fear or it's just laziness, I don't know, but it's there. But as a engineer, I think it is important that we reach out to them, which is why invasive tests needs to be, as much as possible, needs to be eliminated from the system, which is why subdermal devices can take up that space. So cortisons does one thing very nicely, that is painless monitoring. So we are going to create a subdermal device using microneedle patches that will get inside in your skin, little bit inside your skin, not exactly not to the blood capillaries, but to the specifically to the ISF. And then it will sense the cortisol level present inside your body. Now the cortisol level can be spiked for many reasons. If you drink coffee, it can be spiked. If your supervisor is kind of yelling at you, your cortisol may spike. But my particular target is not for everyday human. My particular target is for people who are already facing some travel and inside an ICU. Why? Because ICU can be thought as a unit cell for monitoring the health conditions. So when you are checking for different vitals, let's say heartbeat and SpO2, cortisol can give you an extra impression of what is the situation of the body. You are administering lot of drugs, and as a patient, you probably don't have any idea what drug is going inside you. And most of the cases, one of the recent cases that happened, that inside ICU, one patient died and the family members just barged inside and destroyed everything because they thought that drug is not, the delivered drug is not proper for that patient. So this can be one significant bridge between the family of the patient and the doctor itself. If the cortisol, after administering the drug, your cortisol is increasing, that means the drug is clearly creating some problem in your system. Maybe for good, maybe for bad, that's a different story. But that's where cortisol comes into play. So this is the exact schematic that we came across with. We will be using acupuncture needles. Since their tip diameter is very low, it can be very easily inserted inside your skin, and PCB boards will be used as a stationary compound for this microneedle patch. Then for to polymerize it, will be first coat the acupuncture needle with platinums. Then we will be using cortisol specific MIPs. Now there are a lot of studies have been done where people have used different types of substrate, different types of sampling techniques, different types of sensors, which is where the entire research will be, that what type of exact MIP we can use so that everybody gets profit. So here is a tentative schematic that's showing that we'll be using first platinum, then we will go for MIP, which will bind with the cortisol present in the ISF, and that will be we will using DPV will decode the amount of cortisol present in the ISF and to the wellness of the patient itself. It will be a continuous monitoring system where the cassette or the platform can be changed overnight. So meaning 24 hours you can keep one probe and then you can change it. But all this is tentative because the cost and the market is very vast, and also to put one extra device in this part of this country is a bit challenging. There is a lot of things needs to be taken care of. So which is why we have shown the global market, that global market in the developing country. Recently I was was a part of a conference where people are talking that we need to track the cortisol level more often than we do now. But there are few people from Africa and India who said, well, our tests are so low cost, like we can pay 70 rupees and just take a laboratory test on glucose level, PP level and the fasting level. So how are you going to make people wear this kind of expensive sets? That's exactly where the ICU play comes in, because that's where when you get into the department, when you get into the ICU, nobody cares about the money. They want the lives to be saved, which is why this market opportunity is there for India. It will give us, it will help a lot of people, ease out a lot of pain of the family members, and that can be used here as well. So this is our slide. Thank you. We are looking for any questions.
So you've got a sheet of paper here. Did you want to do anything
Yes.
with it? Did you? Did anybody want?
So this is.
Can we display that on the?
Yeah, we can display.
If you leave it, no. If you leave it there, can we display that on the screen? No, we can't. Okay. Live stream. Just leave that there. Leave that there. We'll switch the camera on. All right. So let's quickly summarize. So Group 3, Team of Thrones, talk about CortiSense, which is basically a continuous real-time stress monitoring device, which basically is looking for the stress biomarker cortisol. They talk about 8 billion. They talk about 75% of the adult reports of regular stress. So if you do the maths for a global population of 8 billion, you can do the maths how many people are suffering from the stress problem. You also mentioned another interesting point about diabetics not happy with the blood monitoring, which is another good example where you should move away towards subdermal devices. And as a niche, you've identified the intensive care unit as a niche place where you'd be thinking of applying your products. And you also mentioned about a 40 billion US dollars global market wherein the whole thing works. And you've made it very clear that you'll be using a molecular imprinted polymer. Now, everything you use here seems to be, even your design looks like you're using acupuncture needles, which are costing only pence. You're using a PCB device, which is also looking very low cost because PCBs are low cost. So there's every reason for you to be able to implement this in a larger scale in simpler settings as well, including low and medium income countries. So I don't think why you should just focus on the ICU, but that's a good starting point. So I think for me, everything was very clear, and I think this is basically looking very good on you. I mean, if I would have been one of the investors, I would definitely be interested in investing in this particular project. So I don't have any questions from myself as such right now, but maybe it might come up. In the meantime, I'd open the floor for discussions. Anybody in the audience would like to ask any questions? Yes, Satya.
So cortisol is present in plasma. You can check it through plasma, which is what people do right now. But the ISF is sensor is MIP based sensor. So technically, what polymer will be used? We haven't, because there are lots of polymers that people have used. So that is the question comes: which one will be most efficient according to this current environmental conditions? Although I am targeting for ICU where conditions will be a little bit controlled, but that, yes. Yes, so no, the thing is, sir, cortisol is itself alone is not enough. So what I have thought is making a platform technology rather than just targeting one. But cortisol will be the main, you can say, the front runner of it. So as far as the saliva picking goes, if you look into the COPD patients, they are cursing the nurses like anything because they are so much in pain because of that forceful oxygen. They cannot open their mouth. That's a huge problem. So, in fact, when I was like lying down in the ICU, I didn't want to talk to anyone or put anything inside my mouth. So, these are very practical things that I have observed, which is why I went for the continuous sensing.
Some real anecdotes, right? Real stories here from some real experience, and that's how scientists and biomedical engineers evolve. That's a fantastic way, and I'm glad you've taken all the messages on board. You've taken all the challenges, and I've got your team on board also to work on this one. I think what you're also referring to is in future you plan to have a multi-analyte sensor,
Yes.
so that you can target. Do you have any idea what would be the other analytes you would like to target?
Sodium would be the first one.
Sodium ions.
Sodium ions.
So for the sodium ions, what would you like to do? What would you use? What would be your sensing principle?
So far I have thought about using continuous system only, but using normal sodium-based electrochemical sensors.
So ion selective electrodes.
Ion selective electrodes.
There will be a challenge because you're integrating two different types of systems.
Yes.
You can actually integrate, you can use molecular imprinted polymers for metal ions as well in principle.
Okay.
But you have a problem there that if you use two different systems, then getting the two systems to work together on the same platform, that's going to be a bit of a challenge there. Pardon? Exactly. So there will be some serious challenges of manufacturing. Any other questions? Anybody? No? In that case, can we all give a big round of applause to the team of Thrones? Thank you so much. All right, can we have our next group, group 4? Are ready? What are you calling yourselves? Are you calling yourselves the Super Kings or Super Queens? Sorry? Avengers. Okay. So we have the next group, which is the Avengers. After this, we've got two more groups, yeah? No, there's one here, and there's one with this. So two more groups. Is 3 groups, yeah? You boys are left as well? Okay, so 3 groups. Are we ready? Okay. So we have group 4, Avengers. Can I please request everybody to maintain silence and show some respect to the presenters, please?
Good afternoon, everyone. I am Vivian. Okay, I am here to present the introduction of our project. Our title is Subdermal Continuous Monitoring of TSH using MIP based biosensor. First of all, around 200 million people in the world are affected by the thyroid disease. Thyroid disease is a disease causes when the abnormal TSH, when thyroid gland secretes abnormal amount of TSH. The traditional detection of thyroid levels are serial ELISA method and invasive, and it's a discontinuous method. Our plan is to make a device which needs a continuous detection of thyroid levels in real time and less invasive method.
Our team has also come up with the idea of integrating MIPS into the sensor, where the MIPS are can be made in different methods. Like the template will obviously be the TSH, and the functional monomers like acrylamide will be binded with the template in hydrogen bondings, and then it will be cross-linked for stable molecule to be formed, and then it will be polymerized for the stability, and then the template will be removed. We are also planning to integrate CGM sensor along with the near field communication system, so that you can be able to monitor the TSH level using a easy application on your phone.
So for this purpose, I am discussing about a type of designing patch kind of thing, so that we can introduce it by for sensing the TSH level in the ISF fluid. So the materials I will be use in this case, like a triboelectric layer which contains the top layer act as a human, which is the human skin. Then the middle layer, which is a TiO2, which is a photoelectric layer, and also that the PDMS. All there has their respective roles, and after that some electrodes which can create some electrical stimuli onto the device. And then I am not using the micro needles here. This is the material I selected for a different type of design, so it is more similar to that. And the conductive polymer like polypyrrole, which creates a hydrogel like hydrogel, and which contains some the MIP embedded into it, and then some bioadhesive for the attaching these devices onto the skin. So the first picture is basically I designed this picture for the wound healing, for the deep tissue wound healing, where the releasing of the growth factor in a controlled manner. But in this picture, you can zoom it. This picture, this one. So in this picture, I used 3 layer like TiO2, PDMS, and our human skin will act as a positive layer. When we switched it on, then it creates some electrical field due to the difference of the triboelectric series. Human skin will act as a positive layer, and PDMS act as a negative layer. And TiO2 is a photoelectric layer which can be activated and increase the electron density by the light, like sunlight or any kind of UV light, etc. So when it is switched on, then the electrical stimuli passes to the electrodes, which is made up of graphene, and then it will degrade the layer which contains the MIP layer by layer. Like if you not use this layer by layer fashion, then the more release of the receptor and the TSH interaction will create a high amplified signals. That's why it will become problematic. That's why I am using this layer by layer, and the TiO2 and the MIP antibodies are MIP basically embedded into hydrogel. This hydrogel contains polypyrrole, which is electrically conductive, but the concentration is different because of the because if we use the same concentration, then the same electrical stimuli can be degrade all the layers at a once. That's why I think this device can work for a 1 month, and every 7 weeks we need to switch it on this device, so that we can go from the first layer to the second layer, and the MIP will expose. Then after the MIP is exposed, it will interact with the TSH, and after that this sensor will sense these interactions with MIP with the TSH and get a signal, and we can get the result. We can also introduce NFC for sensing NFC, which can directly connect with our phone, and we can real time monitor it. So this is the device kind of I made in couple of minutes, and the switches function, the switch on off function can be easily
easily can be done by the patient by its own, and the graphene coating is electroconductive, that's why it is more feasible, and polypidol on sand is it is electrically conductive, and also it can provide mechanical stability. So this is the I made this device idea, and then
Okay, I just want to sum it up, like why we choose TSH for a sensor, that to a subdermal biosensing device. So, since we've been said that around 200 million people around the world are affected, 200 million people around the world are affected from thyroid. So the most common way of diagnosing the thyroid is via blood test, as since it's more accurate. But we can also see that there are reports that in ISF also there is like around 0.4 to 4 milli international unit per ml thyroid hormones TSH levels are there in the ISF. So using that, we can continuously monitor the TSH levels, so that they can know when their thyroid levels are too high or their thyroid levels are too low. So we came up with this idea, and the advantage is that we are using the MIPs so that it can mimic the antibodies, like without having to actually integrate in a biological unit or like biological risks. Also, the advantages are reusability, continuous monitoring, and we can make it cost effective by using like some sustainable reagents and other things are known. And the main thing is the ISF lag and the challenge, which is a major challenge, because according to like the diffusion delays, the diffusion process delays the TSH, like the visibility in the visibility of the ISF in the TSH, visibility of TSH in ISF is a little it's a little delayed as compared to the blood. So that can be done by integrating the AI/ML tools also. The main thing here to focus here is after developing this biosensor, we can then move forward to introduce like hormone responsive drug delivery system. It's not it's actually not a very widely accepted thing, because the thyroid patients, they need to have their medications like via the tablets and all that thing. So we can research on that, like the sensors can we can introduce a kind of drug in that. So that's it for, thank you.
Fantastic. Can we have a round of applause, please? Okay, so clearly they mentioned about 200 million people. I think it must be more than 200 million, because I think every, I mean, at least in female populations as well, there's a lot, there's a big, this whole thyroid, I mean, you might have heard this commonly, right? Oh, this person is suffering from thyroid related problems. This is a major problem, and it's a global problem. It happens a lot more in low and medium income countries, because I think it's got to do something with your diet and as well as the body producing thyroid stimulating hormone. So very well done on identifying a very, very a very critical need. So it's a real challenge there, and so so you're talking about subdermal sensors. Can you go back a few slides, because you've used a term here. No, not this one. Next one. No, you're going backwards. I'll just go forward towards the end. Somewhere you use the term implanted. This one here. Look at this one here. Minimally invasive implanted under skin. Is that deliberate? I think that's probably a mistake, right? Just have to be careful, because when you talk about subdermal, you say more something that can be inserted over the skin, not under the skin. So this thing, you need to be a little bit careful there, right? Because everything was going fine, and then suddenly I see this, and I'm like, probably something has gone wrong with your Google search. It's all right, don't worry about it. I mean, look, we are all humans. the main meaning of being a human means we are meant to be making mistakes. That's fine, as long as you can correct your mistakes, that's fine. So, I mean, from what I understand, you're planning to use molecular imprinted polymers as your bioreceptor system. That's all fine, very nicely shown designs. Looks more like multiple channels. Did you realize that? So you can have multi-analyte sensors, or you can use the same channel to do multiple measurements of the same particular analyte. Are you also planning to bring in any other analytes?
We can also introduce any other analytes like we can sense the glucose by this glucose.
Yes. So what would that be? Anything that's also related to TSH as well. So think about it, because it's nice. I mean, I like the idea of using multiple channels for a single analyte, because that makes your sensor more and more reliable. We don't have a clinician, but one of the things a clinician will tell you is they like multifactorial studies. They like to see what are the things that go up, what are the things go down, and then they bring in the machine learning and the deep learning to get a proper clinical decision support system. So that multi-channel thing is very useful for a clinical decision support system here. So that looks a very neat idea. I think I don't have any questions. You also mentioned about the dynamic range, which was 0.4.
0.4 to 0.16. No, 0.4 to 4.0. Actually, it's in serum, but in an ISH, it's 0.16 plus minus 0.08 milli international unit per ml.
Again, we need to convert that into to get an idea about the dynamic range. But that looks something that's very much doable as well. All right, I don't have any questions. Has anybody got any questions for the Avengers? Yes, here we go. Yes, thyroids.
No, sir, I don't check specifically. I don't know the which can be sensed by the MIP. But if there is various type of thyroid, then we will need to insert different type of antibodies which can be specific MIP specific for that type of thyroid types, and then we can also sense it.
That's a very good answer. Very, very good answer.
We can also change the the MIPs with respect of the.
Feel free to use antibody. You can say plastic or synthetic antibodies. Good. All right. Any other questions for the Avengers? No? In that case, can we have a big round of applause for the Avengers, please? All right. Thank you so much. Can we have the next group? That's going to be group 5. What are you calling yourselves? Sorry?
Smart scale squad.
Smart scales?
Smart scale.
Smart K.
Yes. K for potassium.
All right. So we have the next group 5, which is Smart K. Are these the feedback forms? I hope you haven't done any damage.
Direct HTML में भी नहीं हो ना हाँ हो गया हो गया
It just came a few minutes before.
So good
So we have our
afternoon.
next group, which is group 5, smart case. Over to you.
So good afternoon, everyone. So we will be presenting about subdermal potassium sensor, and our group consists of Rishita, Shubhrati, me, myself, Tushar, Shomojit, and Ayush. So basically, potassium is one of the most critical electrolytes in the human body that is found, and it is essential for maintaining your nerve signaling, the normal heart functioning, muscle contraction, and everything. So when we have more of potassium levels, that is more than the range of 5, so millimolar per liter, it is called hyperkalemia, and when we have less of it, it's called hypokalemia, that is less than 3.5 millimolar per liter. And when we have low hypokalemia, hyperkalemia, it can lead to arrhythmias, mainly cardiac arrests and heart attacks. So we have got this inspiration from a recent case study. Everyone, I think, in India knows the death of Sanjay Kapoor, husband of Karishma Kapoor. He got an heart attack due to allergic reactions when he swallowed a bee while playing polo. So this type of cases are very common, and we can see it from child to like even in old age also, the heart cardiac arrests. So that's why we have come up with subdermal microneedle patches for monitoring the potassium levels. And why subdermal? We are not going into details because this has already been discussed many times during the duration of the course. So it is minimally invasive and it is continuous monitoring, biocompatible, high sensitive, and integration with the wearables is of course one of the greatest things. And now how we have made it will be explained by Rishita.
So these are the chemicals required for preparation of potassium sensor. Firstly, we'll be using high porous, we'll have a substrate maybe of gold or any material, which is electrodeless, a nickel gold immersion electrode, or PCB electrode also you can use. Then on the top of that, we'll be using, we'll electroplate with using highly porous gold nanoparticle. So the structure will be like this, as seen in the figure. Then we'll deposit P dot PSS for the good electron it acts as a good electron transducer. Then we'll use the simplest drop casting method for the preparation of the potassium electrode. So these are the steps followed for the PVB reference electrode. And these are the steps that we followed for working electrode of the ion selective electrode. Potassium is an ion selective electrode. So, and for calibration, we'll use open circuit potentiometry. And the noise response we expect to get is around 59.2 millivolt. So in open circuit potentiometry, we used mainly 2 electrodes, that is the reference and working electrode. Working electrode is the ion sensitive electrode, and reference electrode we have modified the AgAgCl into polyvinyl butyryl reference electrode to remove the potential drift. And AI integration will be explained by Somyajit.
Okay, so now we have the data. Okay, so now that AI term is kind of a overused term actually, right? There is a new term called AGI.
Can I use the mic, please?
Achha. There is a new term called AGI. What is actually AGI? AGI means that artificial generic intelligence. Like in this case, we are designing some sensors, right? Like there is some clinical data was also there. Let's say for 10 people or 50 people, let guess 100 people, okay? And we are generating some data, extracting some features from different aspects, different values from the sensors. But it is not possible to design a sensor for a huge number of chunks. Let's say in Rourkela, we are designing some sensors for Rourkela people. But maybe that sensor is not working well for different parts of India, like Chennai, Kolkata, or different parts. Okay. So right now there is a few techniques as there, like semi-supervised approaches. What actually that semi-supervised approaches do? They only take a small amount of the data, and they only actually focusing on the feature representation of the data. That's why the feature representation learning is more important when you have very less amount of data. But our task is that we have to predict some a forecast or something, a prediction for a huge number of chunks of data. So that's why
in future maybe, or maybe like in recent years also, maybe for sensor designing also, like instead of only AI integration, we have to use some different techniques. Like इससे क्या होगा, there is another term called domain expertise. Like we need, like medical data are very much costly. Okay, to annotate a medical data is like we need, actually this is a very tedious task, and also we need to hire some domain expert to annotate the data, like this data belongs to this level or something. Okay, so that's why maybe less amount of data is more efficient if we use less amount of data to to bring some good like feature representation. Then maybe in future also, like we can develop some good robust algorithm in the field of semi-supervised approaches. Okay, thank you.
So we will be see what are the market trends for using this potassium biosensor. So we know the need of the personalized medicines nowadays is day by day increasing, because everyone wants to be so much precise for their sensing and personal data. So here we are using the very simple method of dropcasting, and that's why we are having using the robotic arm for the particular process. So we will be cutting out the cost very much, and it will be subsequently the most cost effective and reduced cost sensor. So these are the why we are using the dropcasting as a robotic arm. It's a competitive edge technology, so we will get the higher precision. It will be helpful in scalability and cost reduction, and we will be using high quality control. So it's used in the consistency in manufacturing and leads to regulatory compliance and higher costs. That's it. Thank you so much for listening.
Okay, Prof. Fantastic. So that's another nice piece of work here presented by the group, Smart Case Squad, on subdermal sensors for monitoring potassium. That's all very clear. I think you've identified a very good niche here, wherein basically you're talking about hypo and hyperkalemia, which is a problem related to high and low levels of sodium. And you're introducing a single analyte sensor, which is based on the bioreceptor valinomycin. That's going to be your inochrome there. And you're using a P.PSS system. And then the nice thing, the coolest thing you've done here is dropcasting. So we discussed about this morning. If you're able to dropcast things, if you can automate things, you can make them scalable. That's basically part of your scalable technologies. And then you mentioned about using open circuit potentials. That is also a very easy way of implementing it in commercial samples as well. And then you mentioned about the AGI thing, where you mentioned about the artificial generic integration. And of course, then Tushar mentioned a little bit about the personalized medicine side of things, where people can monitor their own glucose levels. These things are really useful because measuring potassium in response to therapies is also something which requires continuous sensing. So you want to basically decentralize the whole treatment aspect so that you can move things from the hospital to more kind of home settings as well. That's fantastic. So I think it's very clear to me what you want to do here. So everything looks very good presentation as well. I don't have any questions for you. Does anybody here have any questions for the group? Prasoon? The anatomical location.
Like your arm or forearm and anywhere. And we will reduce the size of the microneedle also, because it requires only 2 electrode. So maybe how much? 1 centimeter. It would be hardly 1 centimeter. Yes. Yeah, we can. So firstly, this is ion selective electrode. So it will, I think, as for my knowledge, it will, like valinomycin is the ion selector for potassium only. So I don't think, and since we are using PVC also, so other interference will be reduced by the thin layer of PVC.
Yeah, I mean, I agree that the ion selective electrode strategy for building that ion selective electrode is getting the specificity for the particular INO4 that they're using, that's perfectly fine. But I think one of the important things that he raised is about multi-analyte sensing. And I think this is something we also mentioned last presentation as well. If you are able to do multi-analyte sensing, if you can measure sodium and potassium, because as I said, one of the things, we are all developing products which have to be approved by the clinicians. If the clinicians don't approve it, this product is going nowhere. And what clinicians like to see is something which is multifactorial, right? If you're talking about cancer, they would like to see multiple biomarkers there, and they would like to see a lot more data to make it, to show that the sensors are reliable. So maybe you can think of other, for example, when you talk about people with heart diseases, you may also want to monitor things like troponin levels and stuff like that, or myocardium and things like that.
Yes,
so
yes.
that's a good thought. So maybe
actually
you can.
we wanted to reduce the size of the electrodes, so we thought like one analyte will be better.
Well, when it comes to a patch, I think there's no difference between 1 millimeter and 1 centimeter. Simple as that, the patch. Because I think if you speak to the engineers here, if you speak to Khalid, he's going to talk about things like bioinstrumentation, trying to integrate everything on the back of it to get it similar to your this thing. So I don't think you've seen this. You've seen the actual design of a CGM sensor. I don't think you can go any smaller than that. If you go smaller than that, if you don't have the real estate to put your electronics on the back of it, it makes it much more harder.
Yes.
So miniaturization is fine, but don't try to make them so small that they, because think about something which is 1 millimeter small. How are you going to pick it up? Right? You're not going to use a tweezer all the time. And that's the difference we find between working in a clean room where you work in very small samples, but those things have to be stuck to something which is a bit more larger. Right? So think about that as well. Yeah, but very well done. Excellent. Good job.
Thank you, sir.
Another round of applause, please. That was group 6, I believe, or group 5. Can we have the next group, please? Are we left with? Are you using a MacBook or is it a Windows system? Let's try and see. If it doesn't work, then we may have to request. All right, can I once again request, especially for those of you who already finished, if we can maintain some quietness and show some respect to the presenters, please. As you all appreciate, there's a lot of effort that goes into these kind of presentations. Doing a presentation alone is very easy, but doing it in a group is the hardest thing you can do, because you've got to, convince others as well and come up with what we call as a common minimal plan. So let's show some respect and let's maintain some silence so that we can all hear the presentation clearly. So the next group we have is group 6, and they call themselves as Raurkela Challengers.
Good afternoon, everyone. So here we are going to present you regarding about the hormone serotonin.
So we are going to hello. Okay, okay, okay. It already started April. So start. So good afternoon, everyone. So here we are going to present about this serotonin, which is
Sorry, Jerry, let's just have to interrupt you. I think we lost the signal.
So we are going to present about serotonin hormone, which is considered as regarded to the mental health issues and regarding the issues. Basically, serotonin is a neurotransmitter hormone involving in the about human sleep, mood, appetite. The irregularities in this hormone regarding to the serious issues in the human being, like considered as a disorder regarding to depression, sleep.
Sorry, my sincere apologies. I'm having to interrupt you again. We can't see anything on the screen. Is there a chance we can quickly help them with?
Just exit the slide. You don't touch it. Just exit the slide. Yeah, just leave it like this. You can present it. Okay. And you can see it now. Actually, what you have done is duplicate. Yeah, make it duplicate. This is you have this. This is wallpaper, right? One. Move this. Where is your monitor? Mouse. Actually, you should do duplicate the displays. Okay. So you have this.
The next group, whoever is ready, can you please have it ready on a flash drive so that if there's a problem with Windows, we'll request.
Extend for it.
You don't have flash drive?
Display settings.
It's a MacBook. That's fine then. Have you got this on a flash drive? On a flash drive? On a memory stick? Huh? No, it's laptops are not working. Only MacBooks are working for some reason. Has anybody got a spare flash drive, memory stick? Rishita, आपके पास memory stick है क्या? Have you got a memory stick?
USB. Right.
USB drive. है? दे दो इनको. Can I request you? Sorry for bothering you.
Start the presentation.
Here we gotta save it in.
Just ring that.
Give it to him so you can use his.
WhatsApp पे जाएगा फिर वो उसको भेजेगी
होगा होगा उसमें तो मेरे पास है हो जाएगा खाली एनटीपीएस में फॉर्मेट चेंज नहीं किया होगा तो हो जाएगा ये फोल्डर बना देंगे लैपटॉप इतना पता नहीं स्लो भी है
It's not coming through the WhatsApp?
No, sir, network issue again.
Okay. All right, we're going to resume. We had a bit of a technical glitch here. The systems that we have seem to be a little bit biased towards the MacBook systems. They don't like Windows for some reason. Yeah. Can we start from the beginning? Yeah, please. So, thank you everybody for your patience. Thank you so much. Really appreciate it.
So good afternoon everyone. Here we are going to present about serotonin detection for mental health monitoring. Basically, serotonin is a hormone which is a neurotransmitter hormone involved in about human sleep, human mood and appetite. If there is an issue regarding the serotonin hormone in the body, it may trigger some serious disorder like related to the depression and sleep issues. So monitoring this serotonin hormone in real time is big challenging and it's very important to current generation, because in current generation every human being is very fast being work in very workload. So being evolving the stress condition is very easy in this generation. So monitoring this kind of hormones regular basis or real time basis is important. So next, as I said, serotonin is a neurotransmitter function like regulates mood, sleep and appetite. It impacts many critical body functions. So imbalancing 5-HT are linked to the depression and cancer related issues, and understanding these links are very crucial and important in today's generation. So continuing monitoring is vital. It aids diagnostic and neuroscience research. It drives some new discoveries for regarding the sensors and all these things. So what are the overcoming problems we can think to developing a real-time monitoring sensor, which is for current generation there is no real-time monitoring system available. We have to do for in situ analysis, required offline sampling, lack of immediate feedback or immediate results, and bulky and very lengthy and expensive procedure. So for this overcome this all issue, we have think or we just come up with the idea how can we solve this issue using the real-time sensor. So basically we are using a aptamer based sensor. So basically aptamer is
So basically, aptamer is a small peptide sequence or single stranded DNA sequence, and they are very specific to the any protein, any hormones. So they bind to specific targets, such as, like for our example, we are considered as a serotonin to ensure the high accuracy. So their binding is we are considered as a real time or continuous real time binding. So we can detect the how serotonin is reactive to aptamer and the changes in the voltammeter. So we uses here, like we think we can use 4,4 aptamer, and it's showing the strongly affinity towards the serotonin. So next is like how we have like basic mechanism overview of how it will be work. The basic, it will be the aptamer bonds to the gold electrode. Like we are thinking to taking a gold electrode and binding to the aptamer to gold electron using the thiol gold chemistry. So it will bind to the electrode. Second is serotonin will bind to the aptamer, and this induces the conformational changes, and it alters the molecular structure. Like it changes in the electrical impedance, and this change in or increase in the electrical impedance can be detected using the sensor. Basically, single frequency electrochemical impedance spectroscopy sensor we are using here to change to analyze the change in the detection. So next is like conformation shift after impedance, and this affect the electron transfer, and this change we can easily detect it. So here we can use the single frequency EIS sensor or the square wave voltammeter sensor, but for precision single frequency EIS sensor we can think of use. So next is like what are the experimental design, why we are using all these things, because one part of this is a micro scale chip basis which is do the analysis things. Second is like we are using gold working electrode, and then the platinum counter reference electrode, and ensure the accuracy range. And for sample analysis, we taking sample from ICF using the hollow microneedle of size of around 80 micrometer, and the hollow needles are these hollow microneedles are attached with aptamers, which will like insert in the ICF and detect the real time serotonin in the by the reaction, and the changes or difference in the impedance will be detected using the sensor, and these changes can give us about the how serotonin is going in our body. Will it increasing, will it decreasing, in which cases, in stress condition how it is increasing, what will be its range. So all these parameters we can get in the real time conditions. Yeah, thank you. This is the basic overview of what we have thought regarding this sensor.
Right, fantastic, well done. So basically what the group 6 Rourkela challenges are presenting, they're presenting serotonin monitoring here, right? Serotonin monitoring for mental health, a big problem. I mean, and this problem is getting more and more acute. It's precipitating over time, and this is all thanks to the phones that we have. You know, everybody's always hooked up to their phones. This is all that electronic stuff. I mean, I can give you an example from my own personal life. Earlier, we used to do, during my times, we used to do sports for physical fitness or physical health. But these days, sports are more for mental health. I mean, that's the level of so I get both my boys to play, to go outside the house so that they're not on the PlayStations and the Xboxes that people are naturally addicted to. And the problem with electronics is it's addicting. So very, very well chosen problem here of mental health. And of course, serotonin is one of the biomarkers of mental health. We also saw about cortisol, which was presented before. Serotonin definitely is a big problem there. So I like the approach that you've used here, a bit different from what others have used. While most of the people have decided to go with MIPS, you decided to go with aptamers. You've even given a name of the aptamer, which is again another synthetic molecule. It's a 44-mer aptamer with a KD value of 87 nanomoles. Would have loved to see some information on the dynamic range of serotonin in the skin interstitial fluid, right? Another interesting approach they've taken here is rather than using solid microneedles, they're resorting to using hollow microneedles. They're extracting and they're doing the electrochemistry in the lumen of the hollow microneedles, which is another smart approach, which is normally employed by people of the likes of Joe Wang. I think that's something. If anybody's interested in apta sensors, especially your group, since you presented on this, the person you should be following is Kevin Plaxico. Right, Kevin Plaxico, again, another very, very big person when it comes to using aptamer. He's done a lot of animal studies as well. So his research is not just about doing things and keeping them in the lab, also taking them forward as well. And then you mentioned about hollow microneedles for use of extraction of ISF. Would be nice to kind of see if you can get the dynamic range, and also to know if serotonin levels in blood and skin ISF also follow each other very, very much. I don't have any more questions. I will might ask you a few things towards the end. I will give some comments towards the end. In the meantime, let's get people to ask the questions. Where are the usual suspects? Where's Prasoon gone? Now, in the absence of Prasoon, I think I'll have to ask Khalid to ask some questions.
Yeah, I think the serotonin is present in the
it is present in the ISF, but level, how much level it is present, I haven't looked after that, so that I will see.
So we refer one of research paper. They have not developed a completely real-time monitoring, but they have just mentioned they have developed a device that for serotonin analysis detection. So we just modified. Can we do it like for real-time basis, or like our knowledge of information, so we make it make sense?
Yeah, so that's a good presentation. Again, I would ask you to, I'll rephrase it. I would request you to also think on the lines of multi-analyte sensing. It's the same thing that we talk about if you want to convince the clinicians to use it in practice.
So actually, I was in my thinking, I was basically try to thinking of developing a detection kit or real-time monitor kit for depression. Basically, as you know now, depression is a leading cause of
Yes.
deaths,
Yeah.
after cancer and other
Yeah.
diseases. So mental health is very important. So also for depression, cortisol and other hormones are also responsible. So I will think to channelize those hormones also with this if it is a success.
Why not? Why not? I mean, you have to think and you have to come up with new ideas. And the best people to advise you on what would be the best combination would be the clinicians there. All right. Well done. Excellent. Thank you so much. Another round of applause, please. This is the last group that we have. Group 7. I think they have a MacBook. Group 7, what do you want to call yourselves? Surakshak. All right. So the next group, last but not the least, is our group 7. They call themselves as the Surakshak group. Surakshak, and they're going to be presenting on headband wearable biosensors, basically something that's going to help the miners. Can I please once again request everybody to maintain silence and pay attention to the presentations as well, and show respect and the kind of attention that you expect for your own presentation. Can we show the same kind of presentation, show the same kind of attention as well here? All right, over to you.
Good afternoon, everyone. Thank you for your patience up till now. So, as we know that Odisha and even India is a great industry for mining. So here we are with a headband wearable biosensor that detects lead in miners. So let's move forward. According to the reports from UNICEF, as we have seen that over 100 million adults in India are having chronic diseases that is related to health hazard and neurological, kidney and cardiovascular issues. There are many economic impact as well because the working population is working in this area. Current gaps in monitoring this is that it is really slow and very infrequent. Invasive bloods are taken for bloods for sampling and all that stuff, and real-time data is hard to collect. So here we are introducing Surakshak, which is a comfortable wearable band that you can wear around your forehead because it collects sweat, and it can be easily protected using a helmet that is worn by many miners. It is non-invasive, and real-time alerts can be given if the amount of sweat is less or more. So the sweat collection thing is really easy. As you can see, the design is really simple. The biosensor array electrodes react with lead ions and create measurable electrical signals. Data transmission is done via Bluetooth, and it is wireless. Analysis and alerts are easy to detect. So before moving forward, I would like to ask a question from the audience, like what can be used as ionophores or nanomaterials in your biosensors to detect lead? What do you all think? Any answers? Anyone? So we will move forward.
Now we will move to the mechanism of how we are creating this. For first of all, the first step is EDTA has high affinity for divalent metal ions like lead, and it forms a strong coordination complexes. To function like graphene oxide, EDTA and thiol groups will be used for lead ion specificity. Covalent bonds of EDTA to the carboxylic acid and epoxy groups on graphene oxide by a amide bond. Now, I would like to show some data. Concentration of lead in our sweat. Environmentally, occupationally exposed people have 2 to 50 microgram per liter, or we can say parts per billion. And highly exposed people may have 100 microgram per liter.
Now let me walk you through the materials and components used in constructing the wearable lead biosensors. Number one is graphene oxide. We use graphene oxide as the base layer of our sensor. It provides a large surface and great electrical properties for detecting signals. Number two is lead specific ligand. For example, EDTA or thiols aptamers. We attach lead specific ligands like EDTA onto the geosurface to ensure selective detection of lead ions from sweat. Number three is coupling reagents. Example is EDC or NHS. We use EDC and NHS as coupling agents to bond EDTA onto the geosurface through amide bond formation. Number four is electrode substrate. We use substrates like SPCE or gold electrodes to provide the base for the sensor's electrochemical reactions. Number five is reference and counter electrodes. Example is Ag or AgCl, carbon and platinum. Reference and counter electrodes ensure we have a complete and stable electrochemical system for reliable measurements. Number six is buffer solution. We use buffer solutions like PBS to mimic sweat conditions and ensure a stable environment for lead ion detection. Number seven is potentiostat. A potentiostat reads the electrical signals generated during lead detection and transfer the data to a connected system.
So I will be talking about the impact that impacting life enhanced miner safety and productivity outcomes. So as we know, the device name is Surakshak. So Surakshak will reduce the chronic lead exposure in miners, improving their health and productivity while saving healthcare costs for employers and helping miners to meet safety laws. So as you can see, our solution significantly enhance miner safety to improve health, increase productivity, cost saving, and regulatory compliance. So these are the reference we have used, and thank you.
Well done. Can we have a round of applause, please? I think this is a very, Khalid, you've got to support me on this one. I think this is a very good idea wherein you're addressing a big problem, which is a problem associated with the local need of the local community as well. We are in an area which involves a lot of mining activity. So very well done on the research, very well done on the identifying the problem. So I really appreciate it. And you already mentioned there are 100 million adults in India who are minors and who have this problem of chronic exposure to lead this thing. Again, another nice idea wherein they've moved away from the typical subdermal devices to more epidermal devices relying on sweat samples. Very good design that you have of a headband which goes with this thing. So that is also very, very neat. You collect sweat and then you have electrodes that react with the lead ions and you have ionophores there. Everything looked fine. There's only one concern I have. Any interference? Would you expect any interference? So you're using EDTA in this case as a, I wouldn't call it a bioreactor. EDTA is a natural, it's a ligand. And EDTA will naturally bind to ions which have got a 2 positive charge. So one of the possible interference, in my opinion, would be calcium. So if you have calcium in the sweat along with lead ions, it may not be the case. It may be the case wherein they have more lead ions there and a little bit of calcium. Then you will again have to bring in your machine learning and AI algorithms to compensate for those kind of interferences that you may see because of.
It can also depend in the mining condition also. Now, if the miner is very like down to earth, then
yeah,
the sweat condition will there will be a lot of lead.
There'll be a lot of sweat there as well. Yeah, I totally agree. I mean, I'm not denying any of your research that you've done here. And the two concerns I had was, one is on selectivity. You know, there may be some interference and specificity, of course. And then, of course, the concentrations you're talking about parts per billion. Have you seen any evidence of measurements done at that level? Have you got any? Because you in the end you showed 3 papers, right? Can I just quickly have a look? See here. Look at the concentrations that's mentioned here. Lead concentration in sweat of exposed workers was 22.3 micrograms per liter. Now that's definitely much more higher than the parts per billion thing you're mentioning there. So, you know, I think different areas will have different exposure levels. So your biggest challenge will be trying to get that huge dynamic range that you need. So again, you will also need a nanomaterial system at the base to increase your binding sites. So that's another thing you might want to consider as well. And then you also have the first paper which is talking about 0.2 to 25 micrograms per liter, right? That's again, and you're looking down at very picomolar concentrations there. But parts per billion is very, very hard. So that's good. But I like the way you like the choice of the problem that you've mentioned here. The problem is definitely an acute one, addressing the needs of the local community as well. And well done on choosing, on doing something different here, on using epidermal wearable devices there. So well done, excellent, good presentation. I'm going to open the floor for questions. We'll keep the applause for a few minutes. Has anybody got any questions here? Or is everybody hungry and wants to just rush towards to grab their lunch? Any questions, anybody? No? In that case, yes, we've got one question from.
We have this primarily on that, but we can think about it.
Yes, we can. Yes, sir. Yes, sir.
Yeah, and then again, it brings us to the same question that we've been discussing with everybody. Why single analyte? Why not multi-analyte? And if it's multi-analyte, why not look for the other? Which would be your other analytes you would want to look at? You can look at other heavy metals, right? Because the advantage you have, depending on the charge, the ionophores that you're using, they depend a lot on the charge. So you can have ionophores which are specific more. So if you have 2+ charges, you can go for lead and calcium. If you have something else, you can go for the other heavy metals as well. All right, no more questions. In that case, can I request you for a big round of applause for the presenters? And once again, a big thank you. A big thank you to all of you. Actually, it's giving me a very good idea. I think for the next GIAN thing, I think we should get rid of all the theory lectures and do only a simple thing. All the ideas that you've done, we should give you the opportunity to create them yourselves and have that feeling as well. And actually, there is a program in Europe wherein they invite, it's basically done in the Netherlands, where you're invited for a competition wherein you build your own biosensors based on your own ideas. So we will provide you with all the materials. and you can come and build your own biosensors. So if you have some good ideas, especially this, the proper, problem, the local problems and stuff like that. I mean, all the problems that you've mentioned here are all relevant to the modern. There's nothing which I don't see wouldn't impact. So if we can have ideas where you can develop your own biosensors, not learn. I mean, you've already learned it now, but instead of just presenting something which is more superficial and hypothetical, if you present something which works and if you can show it how it works and test it, that will be fantastic, right? So I think that's a nice idea maybe for the next GIAN thing, wherein we can have something where we can build our own biosensors there. Also kind of relates to, so this was just to give you a bit of a flavor as to how the whole biosensing technologies work and how they can be adopted for wearable and epidermal sensors. But I'm really motivated and very encouraged by the response I've had from all of you, and I hope you all found it useful as well. Great, thank you so much, and for all the presentations. Thank you. I think we can call
Ready? Ready? Yeah, please ready. You give that. Give enough space so that people can get. Then reading on Manas, our photo by photo. सर मैं थोड़ा सा कॉल कर देता हूँ, आप थोड़ा सा कॉल कर दीजिएगा। ठीक? मैं इनिशियली कुछ
नेम जैसे है उसी तरह की कॉल लिस्ट है,
कुछ कॉल लिस्ट नहीं है, ऐसे कॉल टाइम में। and uh, and um, and my respected faculty members of the dais. So welcome all to the valedictory function. So we are at the flag end of the workshop, a 5 days workshop. It's been a very long and intense and I hope enjoyable experience for all of you. So I would like to take this opportunity to thank Global Initiative of Academic Networks or GIAN program, an initiative under Ministry of Education. I would like to thank our director sir, Professor K. Muhammad Rao, and the administration of United All Club for enabling this whole program and supporting this program. I would like to thank our HOD, Professor Devendra, for his full support for this and always being available for if we require any assistance during while conducting the workshop. And finally, I don't know how, I don't have words to thank enough our respected faculty, guest faculty who has joined us from all the way from the UK, from Professor Sanjiv Sharma. So I just wanted to take this opportunity to show the dedication of Professor Sharma. He has been very helpful, very engaging, and he has given his full time to us. And I would just wanted to highlight also that he was not well on Wednesday. So we thought that, okay, we can rearrange the schedule based on his health. So he said, no, we should go according to the schedule and we should not rearrange. And at that day, he took 3 lessons or 3 lectures. And also on Wednesday night, actually, since Thursday, we have the demo, hands-on experience. So he was in the lab. He bought some devices from UK, sensor devices, sensors which we used in the hands-on experience in the next day. So he was in the lab testing them, making sure everything is right until 11 at night almost. So I really thank you, sir, and I hope that this is the beginning of a conversation and collaboration for many, many years to come. Thank you very much. I would like to thank my co-coordinator, Professor Prasoon, Without his support, it would have been very, very difficult. And he's been very, very supportive, and he has accommodated all my requests all the time. So I would like to thank him as well. And I would like to thank our volunteers, student volunteers, Ruchira, Rishita, and Subradruti, Tushar, for helping out with the live demo and the experimental sessions. And I don't know, I mean, you guys have traveled from all the way from all over country and you are here from, whether you are here for internship and you got an opportunity to attend the workshop, or whether you are attending it just on your own. I really thank you all, and you have been a fantastic audience. And I don't know, the enthusiasm that you have shown, I think even Sir was appreciative quite a lot, that okay, he has never seen this kind of enthusiasm in students. So it's really, really something I should actually congratulate you, and I'm very happy to have you all here. So with this, without any further delay, I'll call out the names and you can just please come to the stage and collect the certificate. There is no alphabetical order, so you just please, yeah, one more thing, I just forgot, sorry. I would like to ask HOD sir to present a gift of appreciation to our guests, please, before we start. Thank you very much, sir.
Just like to add, this is a token gift. This is what we call, this is part of normal formality. I think the best gift I've got from all of you for attending the course so sincerely. I mean, HOD sir, they have been fantastic. They attended the courses, they were answering all the questions, and today we had some beautiful performances where all of them were divided into groups and they came up with some real world problems, solutions for real world problems for global healthcare. And they came up with some really brilliant ideas, and that is really, very, very well, it shows that how much they've been attentive, how much they've focused on the course, and how much they've dedicated themselves to the course. And I'm really thankful to all of you. I think the attention, the dedication, and the enthusiasm that you've showed towards this GIAN series actually is the best gift I've got from here as part of this trip. Thank you so much.
Thank you very much, sir. So we'll start with the certificate distribution. Ma'am, can you please join also? We'll ask you to distribute. Please, ma'am. Please, ma'am. This is a request from us. Please. So HOD sir, you please also distribute few, and a few ma'am will also distribute. It's an honor for us. Okay, the first person is Sagaya Vivian D. Sami. So if you can give, please come, stand here. Yeah, that's it. Please take a photograph, please. Thank you. Sukanya Panigrahi. Prachi Chaudhary, not here. Rosie Rani, okay, not here probably. Arpita Pati. Romin Kumar.
Today. A double whammy for you. Yes.
Aniket Patra. Priyanka Thakur. Kiran Kumar Bindani. Please sit down here. Raviraj Patil. Raviraj. Vishal Kumar. So sir will call out the rest of the names.
So Tharini K, हाँ एक्सचेंज प्लीज. पार्थवी किरण साहू, ममीना प्रधान, उपासना महापात्रो, अभिषेक चौरसिया, स्वाति सबनम तोपनो, सुचिस्मिता विश्वास, आदित्य पुशोला, सयंतन विश्वास, जियांशु सिंह, अभिषेक कुमार गुप्ता, श्रुति सौम्य मिश्रा, मालिनी जैन, ठीक है मैम आप लोग कर दीजिए. चिरन्यू बरापत्ते, चिरयू चिरू सुब्रदीप साहू, सत्यव्रत बेहरा, शिशिता शर्मा, आयुष रंजन, रुचिरा चक्रवर्ती, प्रज्ञान रे, सौम्यजीत गायन, स्वागतिका बारिक, कीर्ति केसवानी, तुषार शिर्के, मोनिशा गौरी, लास्ट सुश्री संगीता मिश्रा. पढ़, इतना ही पढ़ा हुआ थे तो किसका किसका बच गया क्या? एक कोई भी इसमें नहीं है सर।
फोटो होने से उसके बाद मिल जाएगा।
ऐसे डेमो डेमो।
So she was also one of those people who stayed a little late in the evening to make sure the workshop was a big success. Very well done. I'll leave it here.
Yes.
And get you from okay.
Thank you all very much. We just take quick photographs and then we'll be done for the workshop. So, ऐसे बैठ जाते हैं, background कैसे? So you tell. ऐसे? So outside light hold on. Okay, we can go outside in front of the department, entrance. हाँ, एक यहाँ पे